Hola, buenos días.
Hola, buenos días.
Hola.
¿Cómo están?
Hola, Denise. Bien,
Bien.
gracias.
Consulta, ¿quién va a apoyar ahora aquí del equipo de ustedes, doctora? ¿Los chicos que están conectados?
Sí, está María Fernanda, Leonardo, Alejandra. Hola, Lizeth.
Para darle el...
Hola, Manuel.
Hola, buenos
Hola,
días.
Denise.
Para que,
Hola.
Adriana, para que hagas coanfitrión a los niños. ¿Nos
Voy.
pueden dejar sus nombres,
Hola, buen
por favor, en el chat para poder hacer los coanfitriones?
María Fernanda, escuché.
Leonardo. Ya. Los que necesitan probar
Paula Díaz también.
el PowerPoint, la compartir pantalla, ahora podremos hacerlo. Después ya vamos a empezar a compartir, a transmitir en directo.
Listo.
A mí me gustaría probar el mío porque tengo un
Ya.
video que quiero ver si funciona.
Perfecto.
¿Tiene audio?
Sí.
Ah, listo. No, para que cuando comparta, comparta con audio.
El video, sí.
El video no tiene audio.
Ah, ok, súper.
Es este. ¿Se ve bien?
Sí.
Sí, lo que noté es que si uso el puntero láser, el puntito rojo no puedo activar el video. ¿Es así o no? No sé por qué.
No que yo sepa, a ver, puede... Póngalo
Tendría
en
que
grande.
en presentación.
Presentación.
Y después, a ver, ahí se podría activar.
Sí.
Pero si prendo el puntero láser, ahí no
porque
puedo.
no le hace el clic. Sí.
Exacto.
Ya, sí.
Ahí no se puede.
Yo lo que hago es ponerle como iniciar automáticamente el video. Cuando uno pasa la diapositiva, se inicia el video.
Pero eso tendría que haberlo programado de antes, no sé cómo se hace.
Sí, pero pues mire, es muy fácil. Puede volver atrás. A sacar la pantalla completa. Ya, arriba del video, del mismo video, de la diapositiva
Arriba.
4, ¿sí?
Ya.
Haga clic derecho. Arriba del video. No tiene que ser arriba del video. Ahí. No lo dice activar automáticamente.
No, porque...
Porque no está en
PowerPoint.
Ya no sé cómo hacerlo en Mac.
No tiene botón derecho.
No tiene botón derecho, no. Ya, no importa. ¿Será?
Tengo que sacarle el láser.
María José, ¿cómo agrego el fondo de ACCDiS? Así como al Zoom. ¿Cómo se hace eso?
Tiene que ir a donde dice iniciar video.
¿Ya?
Al lado hay una flecha hacia arriba.
¿Sí?
¿Tiene el video, no es cierto? ¿Lo tiene abajo en su computador?
Sí.
Ya. Elegir fondo virtual.
¿Ya?
En elegir fondo virtual, dice elegir fondo virtual y sale una flechita con un más.
Con un más. A ver.
Ya. Y al poner el más, dice añadir video.
Añadir imagen. Ya, imagen o video.
No, pero video tiene que ser.
Es que es una foto, la que descargué, una que habías mandado tú en el chat, ¿no?
Sí, pero es un video, no es una foto.
Qué raro. No se me descarga como video. A ver.
¿Se lo reenvío?
Ya, porfa.
Se lo reenvío.
Hola, buenos días.
Buenos días, hola.
¿Qué tal?
Hola, Alejandro.
Hola.
Hola. ¿Quién más quiere probar su presentación?
Yo.
Adelante.
A ver. Ay, ¿qué hice? ¿Se ve, verdad?
Y se ve bien.
Uy, la presentación sí se ve.
Ya.
Sí.
Súper.
¿Qué más? Va a... Ahí le envié el video de nuevo.
Ya, voy a probarlo.
¿Se ve ahí?
Y se ve.
Sí, bien.
Ya, perfecto. Ahí, súper.
¿Hay alguien más que quiera que coloque como coanfitrión?
¿Puedes dejar de compartir, Alejandro, para probar yo?
Ya. Stop share. Ahí está. Súper. Ay, qué buenas fotos son. Está buena.
Es nuestro microambiente.
Claro, nuestro microambiente.
Dentro de Santiago.
Hay un microambiente dentro de Santiago.
Sí. Sí, se ve bien, yo creo. No tengo ningún video, así que...
Muy bien. Solo que tienen que tener, les digo como para futuro, ya hoy día no, ojo que al lado superior derecho se pone su video cuando uno transmite en directo.
Ya.
Entonces, si
acá.
ahí se pierden, por ejemplo, el logo de la San Sebastián se va a perder.
Perfecto. Bueno, será. O no pongo video.
Ah, bueno, sí, esa es la otra opción. Pero no, pero para que sepan qué pasa, eso los directos, como que queda el video ahí arriba.
Ya, súper, muchas gracias.
¿Alguien más que desee?
Me di cuenta que con el puntero tampoco me pasa las diapositivas. Con el puntero láser.
Tampoco.
No, así que
No,
va a tener que...
no hacen ponerle reproducción automática.
No, con la flecha.
¿Dónde saca el puntero aquí? Eso no creo.
En la pantalla, ¿cierto? Cuando tienes la presentación abierta, botón
¿Ya?
derecho, y ahí aparece Pointer Option.
Ah, ya.
Hola, yo estoy compartiendo. ¿Se ve mi pantalla? Hola, buenos días.
Sí, se ve.
Hola, buenos días. Sí, se ve.
Ah, ok. Bueno. Ay, ¿cómo dejo de compartir?
Arriba te dice cómo dejar de compartir en la parte superior en el medio. Por allí.
Ah, ya. Ok.
En 5 minutos vamos a empezar a transmitir en directo, ¿ya? Así que los que quieran...
María José, te mandé un mensaje por si lo pudiste leer.
Sí, te estoy respondiendo.
Ah,
Mientras María José activa las otras cosas por allí.
gracias.
Ah, es que somos dos María José. Sí, nos multiplicamos.
Sí, ahorita me cambié.
No, no hay problema. Hola, oye, yo no me había mostrado, no había.
Hola.
¿Dónde estoy yo?
¿Falta alguien más que haya que poner como coanfitrión o están todos ya conectados? Los que van a ser coanfitriones, la labor va a ser aceptar a todas las personas que tengan nombre de persona para que no sean bots. Y lo otro sería, ahí está el doctor Quest, sería, por favor, silenciar las cámaras, o sea, apagar las cámaras y silenciar los micrófonos de la gente que no le corresponda hablar en ese momento.
Ya, perfecto.
Eso sería.
Por lo menos yo.
Y ir aceptando, porque por ejemplo cuando yo muestro mi pantalla no puedo hacer nada, porque si no se detienen los videos, quedo como imposibilitada.
¿Se presenta con la cámara?
Si desean, sí, si tiene buena conexión, sí, si no, al final queda como a gusto de cada uno, que a algunos no les gusta verse, a otros sí, así.
Ya, ok, ya.
Así que eso. Al final se puso el logo.
Es que lo que pasa es que te mandé un mensajito, María José, y no lo puedo descargar del WhatsApp, el video. Entonces, si me lo puedes mandar al mail, porfa.
Voy.
Gracias.
Entonces yo puedo ir de coanfitrión igual.
Ah, listo. Karen, sí, yo te coloqué. A ver. Ah, no, perdón. Me faltabas tú.
Gracias, Adriana.
Gracias por avisar. Si hay otra persona que me falte,
Ya,
me avisa.
Ya.
me
Sí, me
envía
llegó.
el video.
Ahí me llegó el video.
Igual doctor.
Ahí sí pude.
Ya. Ya, Adriana, empezamos a compartir, vamos a empezar a compartir, así que si quieren, pues, mejor apagar las cámaras por mientras y después cuando iniciemos las volvemos a prender.
¿Tú nos dices cuándo prender?
Sí, yo les hablo por interno.
Ya. Good morning to everybody. We would like to welcome all of you to today's ACCDiS Spring Symposium, where we will hear about cell signaling in cancer. In today's talks, we will first have a series of 3 20-minute talks by myself, by Alejandro Godoy and Manuel Varas. Then we will have 2 short talks by young researchers from our center, Denise Bravo and Manuel Valenzuela. I would like to remind you to please send us your questions through the chat to discuss them at the end of the presentations, where we will have 15 minutes of general discussion. I'm Lisette Layton, Research Associate of the Advanced Center in Chronic Diseases. And now I will leave you with my colleague, Dr. Denise Bravo, to tell you about this morning's presentations.
Okay, good morning. Thank you, Lisette. Okay, the first 3 talks this morning will be on metastasis in breast cancer and melanoma. And then we will hear about the effect of fructose in prostate cancer, and then a talk on exosomes for chemioresistant cancer cells in tumor progression. So let me introduce Lisette Layton. She is a biochemist that studied in the Faculty of Chemical and Pharmaceutical Science at the University of Chile. She obtained her PhD in science in the Faculty of Basic Science, University of Chile. She trained as a postdoctoral fellow at Duke University in North Carolina in USA, and she there performed a second postdoctoral training in Switzerland. She returned to Chile in 1999 and is currently full professor in the Faculty of Medicine at University of Chile and an independent investigator that has been sponsored by national and international agencies, including FEPS, Fogarty, NIH, Millennium Initiatives, Fondesit, Fondekeep, from CONICIT and IP. Dr. Layton today will talk about the molecular mechanism movement of cancer cells and their similarity to glial cells in the brain when they are activated by inflammatory process.
Thank you, Denise. I'm going to share my screen now.
Okay.
Can you see it?
Yes.
Okay. So I would like to start sharing with you just a thought of that metastasis kills more people than COVID-19 ever will. Why is this? Because, because cancer is among the leading causes of death worldwide, and almost 9 million people die of cancer every year. 90% of these deaths are caused by metastasis. This means that about 8 million people die every year because of metastasis. So what is this? Metastasis is, I don't know why I can't see there, sorry. Metastasis is a process where primary tumors get vascularized. They are detached, then the cells start detaching them and migrate to the blood vessel. In a process of intravasation, they circulate through the bloodstream, and then by extravasation, they go to distant places and form secondary tumors. Cancer cells break off from the primary tumors because they acquire this ability to move them. And this is why cell motility is such a key event in the process of metastasis. So cell migration is a process that is coordinated by integrins, the receptors that can interact with extracellular matrices and connect then the actin cytoskeleton to generate the traction and cell movement. So there is this complex that is formed that is called focal adhesion that contains many proteins. One of them is focal adhesion kinase, which is the one that you can see in this short video that I'm going to show you. This cell has been transfected with a green fluorescent protein fused to focal adhesion kinase. And the cell then to move has to form these focal adhesions or points of contacts and in the front of the cell, attach their front part of the cell, detach the end of the end tail, and in that way generate this forward movement. Like a spider in a spider web, if you think of these points of contacts of the spider with its web, it can move because it generates this point where it have to attach and then detach, and in that manner move forward. The cell does the same thing, except that instead of a spider web, it has an extracellular matrix. And so by generating these points of contact with the extracellular matrix, the cell attach and detach and moves forward. So cell migration in metastasis is induced by VEGF, because as I said before, it when the primary tumor gets vascularized, then it generates factors like VEGF, the vascular endothelial growth factor. Also generates pro-inflammatory cytokines like TNF, and these factors activate the endothelial cells. These endothelial cells then express a protein that is called Thy1 or CD90. And this protein can interact then with an integrin in melanoma cells, the alpha B beta 3 integrin, inducing metastasis. So in this interaction of Thy1 and integrin was originally described by our group, but in a different cell-to-cell context, which I'll tell you in a moment. So in 2013, a group in Germany described that Thy1 was present in endothelial cells in melanoma and metastasis, but not in healthy skin or benign melanocytic lesions. You can see here the CD31, which is a marker of endothelial cells that co-localizes with Thy1 only in melanoma and in metastasis. This group also described that Thy1 was important in metastasis because if they use a mouse deficient in Thy1, so a knockout mouse, and injected with melanoma cells, they generated less metastasis than the normal mice. So in our case, the integrin Thy1 interaction that we described was in brain cells. We studied neurons' interaction with astrocytes, which is a type of glial cell. And what we found is that neurons always express Thy1. However, astrocytes do not express integrin unless they are in an inflammatory environment. And so in that case, the astrocytes then reactive and express the integrin, and this interaction takes place and leads to cell migration. So the astrocytes move in an inflammatory environment. If you think of our brain cells, they don't move anywhere. But if there is an inflammation due to damage, traumatic brain injury, stroke, or neurodegenerative diseases, then these cells can move. And this is triggered due to this interaction of integrin and CD90 or Thy1. This Thy1 protein has an RLD tripeptide, which is we described as an integrin binding site here. So during the last 10 years, we've been describing the signaling mechanisms that are involved in astrocyte migration, and the Thy1 integrin binding leads to the oligomerization of the integrins, activates phospholipase C gamma, which hydrolyzes PIP2 in inositol phosphatidyl 3-phosphate. this IP3 activates the IP3 receptor in the endoplasmic reticulum, and calcium is released to the cytosol. This calcium now is required for hemi channels connexin 1 and connexin 43 to open and release ATP. And this ATP then can activate P2X7 receptor and let the calcium from the extracellular medium to go into the cell. All these events we described in astrocytes that are required for their migration. So if it is thinking of the relevance of this Thyroid integrin binding in melanoma progression, we ask the question whether Thyroid integrin engagement is important for migration of cancer cells. And also we ask the question whether the signaling mechanism that we have described that involves calcium, hemi-channel open, P2X7 receptor activation is involved in Thyroid-induced cancer cell migration. So to test this, we use two types of cell lines, a breast cancer cell line, MDA MB231, and mouse B16F10 cells, which is a melanoma type of cell. We incubated the cells in serum-free medium with or without the inhibitors, and then stimulate them with Thyroid. With this experimental design, we did migration assays, so wound healing assay, Boyden chamber assay, invasion assay, and transendothelial migration. We also measured the kinetics of intracellular calcium, the uptake of Lucifer yellow that measures the opening of hemi-channels, and we also measure extracellular ATP. I'm not going to show you all the results. We did this with the both cell lines, and so I'm only going to show you a few key experiments. For the cell migration assays, we use the Boyden chamber assay in which the cells in cancer cells in serum-free medium are added on the upper chamber. The lower side of this insert has fibronectin coated, and the medium in the bottom chamber is without serum. We let the cells transmigrate for 2 hours and then count the cells that went to the other side of the insert. For the invasion assays, the method is very similar. The only thing is that we introduce here a matrigel layer, so the cells have to go through this layer too. And this, therefore, the timing is a bit longer, it's 7 hours. So we checked then in this these breast cancer cells, whether this was beta-3 dependent, the migration of and invasion of these cells. And when we stimulate the cells with Thyroid, this is a fusion protein that contains an FC tag of an IgG1. And we can see that it induces migration and invasion. And the protein that we use as a negative control, which is a trade receptor 2 FC, it contains the same tag, but it's a protein that is irrelevant in this system. The non-stimulated cells behaves the same than the negative control. So to test the dependence of beta-3 presence, we silence beta-3 first in these cells and compare it with cells that were transfected with the control siRNA. And here you can see that the control siRNA doesn't do anything to the cells. They can still migrate when Thyroid is added, but if the integrin is silenced, Thyroid cannot induce migration or invasion, indicated that the presence of this integrin is required for Thyroid-induced migration and invasion. We then measure calcium from intracellular stores, so whether it was this calcium was coming out of intracellular stores, and with fluorophore we could load the cells and follow the kinetics of the calcium increase, and we can see that Thyroid increases this intracellular calcium, and this comes from this endoplasmic reticulum lumen, because it will use 2 drugs, 2 APB and sexospongin C, which block the IP3 receptor, then the Thy1 cannot do the activation and the calcium, the elevation of calcium in the cytosol. Next, we check whether Hemi channels were open after this stimulation. And for that, I said we use the uptake of lucifer yellow, which goes into the cell when the Hemi channels open. With Thy1, the Hemi channels are open, the dye actually goes inside. But if we use BAPTA AM, which chelates the intracellular calcium, this signal is lost. We also check the Hemi channel opening in the presence of these inhibitors of the IP3 receptor, and we can see that the calcium elevation is also blocked when stimulated with Thy1. So this means that Hemi channels are open due to Thy1 stimulation. This requires calcium, and calcium that comes from intracellular stores. We then check cell migration and invasion, and whether this required ATP release via Hemi channel opening and P2X7 receptor activation. To test whether the ATP came out of Hemi channels, as we have previously described for astrocytes, which use the drugs that block the Hemi channel opening, which is probenecid and eptanol, which can block connexins and pannexin Hemi channels. And we could see that the ATP release was decreased. And if we use both together, then the inhibition was even better, meaning that ATP is released via these Hemi channels. We then tested the opening of the Hemi channels in the presence of these blockers, and the drugs actually decreased the lucifer yellow uptake, and the two together decreased even further. And to test the P2X7 receptor activation and ATP release on cell migration and invasion, we used then A-pyrase, which is an enzyme that hydrolyzes ATP. And what we could see is that, again, the invasion migration too were inhibited by this enzyme. And if we use an A compound to inhibit P2X7 receptor, this also inhibited cell migration and invasion, indicating that these two processes require ATP release via Hemi channel opening and P2X7 receptor activation. Then we measured the transendothelial migration, since this is important in the intravasation and extravasation process. And here the assay is again very similar, but instead of a matrigel, we use a layer of endothelial cells in there that the cancer cells have to go through in order to show this transendothelial migration. So to test whether the presence of beta-3 was important, we use cells with silenced beta-3 or with beta-3. And to see whether the Thy1 upregulation was required, we treated the endothelial cells with TNF and checked that Thy1 was actually increased. In this case, at 48 hours is the time that we use the cells for the transendothelial migration assay. And so here we use the B16F10 cells, these are the melanoma cells, and tested the transendothelial migration. So in the absence of TNF, these cells with or without beta-3 cannot move, but in the presence of TNF, where Thy1 is expressed, then with beta-3, these cells can migrate. But without beta-3, they can no longer do the transendothelial migration. And this, we obtain similar results with MDA MB231 cells, the breast cancer cells. And as I said, we did most of the assays with both cells and obtained very similar results. So for the in vivo experiments to test the dependence of beta-3 in this metastatic process, we use these cells with silenced beta-3 or the control siRNA or the melanoma cells wild type. So this is a syngeneic mouse model where you inject the melanoma cells in the tail vein and wait 21 days and do the, measure the black lung tumor mass. So the result of this experiment is very amazing. The tumor mass in the lungs are very clear here in the control cells, but when we have beta-3 integrin silenced, here the tumor mass is actually very little. And if we measure that quantitatively, we can see that the tumor mass in lungs in the control animals are actually around 40 to 50% of tumor mass in the lungs. Whereas those mice that were injected with the cells that had silenced beta-3, they almost have no tumor mass in the lungs. So as a conclusion, Thyroid inactivated endothelial cells binds to integrin in cancer cells. The downstream signaling that triggers includes the calcium release, ME channel opening, ATP release, and P2X7 receptor activation. And these events lead to increased migration, invasion, and metastasis. So with inhibitors of these integrin Thyroid interactions, such as IgD tripeptides, antibodies against Thyroid or beta-3, we might be able to prevent cancer metastasis, and with that, stop the spreading of these cells to the other parts of the body. And in this manner, we might be able to offer to those 8 million people that die of metastasis every day, maybe a chance to live. So with that, I would like to thank the lab, the Laboratory of Cellular Communication, which is led by myself and Dr. Andrew Quest, my collaborator and husband, and the students that were involved in this work, Mariana Brenet, Jorge Diaz, Samuel Martinez, Ramon Perez, and Leonardo Perez. The funding agency mainly has been CONICET for this study via the programs FONDECIT and FONDAP. Thank you very much.
Thank you very much, Lisette, for the presentation. We will move to the next speaker. He is Alejandro Godoy. He is a medical technology from Universidad de la Frontera. He has a master in physiology at the Universidad de Concepción and a PhD in cellular and molecular biology, also in the Universidad de Concepción. He has a postdoctoral training in Roswell Park Comprehensive Cancer Center in Buffalo, New York. And he returned to Chile in 2013 as an assistant professor. And then the current position that he has is an associate professor at the Universidad San Sebastián and hold an adjunct professor position at the Department of Urology of Roswell Park Comprehensive Cancer Center. So please, okay, thank you. Dr. Alejandro Godoy. You have to turn on your microphone, please. We can't hear you.
Okay.
Thank you.
Can you hear me now?
Yes, thank you.
Okay. Sorry. Let me check this. Okay. Okay. Okay, thank you for the presentation. And I'm going to speak today about something we have been working over the last 3 years more seriously. And but it's an area that I have been explored since I was in the undergraduate, since I was an undergraduate student. So we have collected some information over this last 2-3 years that I'm going to show you today. I have to disclosure that I have no conflict of interest, and that most of the experiments that you're going to see today were funded by the U.S. Department of Defense prostate cancer program. So in this first slide, I'm going to show you the cancer statistic, which is a sort of a paper that is published every year in the CA Cancer Journal for Clinicians. Yeah? And the last version, the 2020, showed that prostate cancer is the first cancer diagnosed in adult men, and the second leading cause of cancer-related death in adult men. So it's a very important disease. And in Chile, the situation is very similar as it is in most of the Western countries. 1 out of 6 men will develop prostate cancer during his lifetime, and PCA represents the second leading cause of cancer-related death in adult men. Okay? And what is more interesting for us is that PCA incidence, prostate cancer incidence, increased over the last 10 to 15 years in our country. Okay. The reason for that, we still don't know, but we have some ideas about why this is happening. Okay. Remove this. Okay. So the risk factors for prostate cancers are, you can classify them in 2 types: the non-modifiable factors and the modifiable factors. Within the non-modifiable factors, we have the advanced age. Prostate cancer is characterized for being a tumor that attacks older people, okay, with the median of the age at diagnosis is about 68 years. Black race ethnicity has been, there is several studies showing that there is that people from African American especially develop more prostate cancer in terms of incidence, and their prostate cancer are more aggressive. Okay. Family history, there's a huge link between the family history, and there's some insight in terms of defining the genes that could be involved in the development of this type of cancer. But still, there is a very poorly explored area in prostate cancer. And the modifiable factors, from which one of the most well characterized is lifestyle. Yeah, and when I talk about lifestyle, I'm talking about Western diets, and especially the use of sweeteners from processed food. Okay. So when you talk about diet, you basically, if you could bring this concept to a cellular context, you basically are talking about deregulating of cellular energetic in cancer. This is a picture that most of you know, published by Hanahan and Weinberg. This is the hallmark of cancer. And when you talk about the deregulating cellular energetic, you are talking about, in cancer, you're talking about this guy, Otto Warburg, which was one of the most brilliant, I would say, scientists that contributes a lot in this area. And before this guy, before Otto Warburg, people that study the way glucose was metabolized in different cells knew that glucose was metabolized differentially depending on the presence of oxygens. Okay, so when oxygen was present, glucose was metabolized to pyruvate, and then pyruvate get into the mitochondria to oxidative phosphorylation to be further metabolized to produce 36 moles of ATP per molecule of glucose metabolized. Okay, in the absence of oxygens, glucose was metabolized to pyruvate, and then pyruvate metabolized to lactate, which was extruded from the cells. Okay, and that result in a lower production of molecules of ATP per molecule of glucose metabolized. Okay, so what Otto Warburg said, he was studying cancer cells, and he was studying also hyperproliferative benign tissues. And what he found is that in cancer cell, yeah, and in this special type of tissues, yeah, whether these tissues have access or not to oxygen, they decided to metabolize glucose to pyruvate, okay? And then from pyruvate to lactate, yeah, with less production of ATP per molecule of glucose, okay? So that was something contradictory at the beginning, because people thought that for proliferation, you need a lot of ATP, which is actually not true, yeah? At that time, Barber proposed that cancer was a disease of a problem in the mitochondria, okay, which actually was not true. So with this less production of ATP or molecule of glucose metabolized, so the scientists realized or postulated that in order to achieve those high amount of ATP that cells needed to proliferate, so the cancer cell has to overexpress glucose transporter, okay? And that concept was published in 2 papers back to back in Science, published in 1987 by the group of Harvey Lodis and Ora Rosen. And what they found is that malignant transformation was associated with overexpression of glucose transporter, okay? At that time, they didn't know too much about this family of protein. There was just a couple of them cloned and sequenced. Today, we know that this glucose transporter belongs to a superfamily of protein called the exose transporter family, okay? From which 14 members have been described until now, and they have 2 basic characteristics. The first one is that they have several ligands. They can transport, they have multifunctional transport capacity. Yeah? So they are able to transport different type of exos and some other substrate, like the oxidase form of the vitamin C, okay? And the second characteristic of this family of protein is that they have tissue specific expression. So they are not all expressed in all type of cells. They have a specific type of tissues that every isoform is expressed. For example, GLUT1 is the most ubiquitously expressed in all tissue and is the isoform expressed in the blood-brain barrier, and also is the isoform that is overexpressed in the majority of cancers, okay? So this has been confirmed by several studies. Here I'll show you one of them that we published in 2006, showing overexpression of GLUT1 in different type of aggressive cancer, okay? So what is important of this? Why this characteristic of the tumor cell is important? Well, for several reasons, actually. In terms of translational research, this characteristic was the provide the biological basis to the development of a technique used to diagnose or to detect cancer, which is called the positron emission tomography, okay? In which you inject radioactive glucose to the patient, and if the patient has a tumor which has a lot of expression of GLUT1, has a high capacity to uptake glucose, so then you can create images of this tumor, okay? Like you can see in this picture, you have a primary tumor in the breast of a woman, and then you have a metastasis in a axillary region, okay? But for some reason that people don't understand yet, this technique that works perfectly fine in a lot of tumors, to detect tumor, to stage tumor, and to analyze the response to treatment in tumors. For some reason, this technique doesn't work in prostate cancer, at the point that it's actually not paid by insurance in the United States to create images of prostate cancer. Okay, the FDG PET, PET using fluorodeoxyglucose. Okay, so the reason for that we still don't know, but we started to think about this, and we came out with two hypotheses. The first one was that the cancer cells could have a low metabolic activity, and that made them difficult to differentiate them from the benign tissue because they actually don't have a lot of uptake of glucose, yeah, because of this metabolic activity, which makes sense because prostate cancer, as probably you know, is a slow-growing type of tumor. Okay, and the second hypothesis that we postulated at that time was that prostate cancer could use other exos. That means that prostate cancer cells do not rely on the use of glucose, but can use other exos, like for example fructose, to fuel their metabolic activity. Okay. So this second hypothesis appears to us more interesting, and we started to explore this hypothesis more in detail because there is a lot of evidence that point to that idea. The first one, and one of the most powerful one, is that the consumption of fructose has increased over the last 25, 30 years, yeah, with the revolution of the food industry. Yeah. Basically, the amount of fructose that we eat per day has increased at least 5 times, okay? And concomitant with this increase in the consumption of fructose, also there are several other diseases that have increased too, yeah? Like for example, metabolic syndrome, obesity, and some type of cancer, okay? One of these cancer, obviously, prostate cancer, okay? So the second evidence that we obtained to follow this hypothesis was that we analyzed expression of glucose transporter, the members that were cloned at that time in 2006. And what we found was something very interesting. We took over 100 malignant neoplasias, all of them very aggressive, and we analyzed expression of GLUT1, okay? Thinking that on this idea that GLUT1 was the transporter overexpressing the majority of cancer, as people know at that time, okay? And what we found is that from these highly aggressive tumors, only 50% of them overexpress GLUT1. The other 50% show low expression of GLUT1, okay? So we thought at the beginning, well, probably they don't overexpress GLUT1, but they overexpress other glucose transporter. And then we analyzed the other glucose transporter expression. And what we found is that from the 50% that express low GLUT1, low levels of GLUT1, they mostly overexpress GLUT2 and GLUT5. And as you remember probably from the table that I showed you before, both transporters have one unique characteristic. They are both able to transport fructose. And actually, GLUT5 is a pure fructose transporter. So it only transports fructose, okay? So with that idea, we started to explore this hypothesis in prostate cancer, and we analyzed expression of all of the isoforms that were at that time reported to express or transport fructose, okay, in benign and prostate cancer tissues, okay? And concomitant or according to the idea that prostate cancer are not able to uptake glucose, we couldn't find overexpression of GLUT1 in prostate cancer when compared to benign, okay? Same happened with GLUT2, but when we went to GLUT5, we found expression in the benign prostate and in prostate cancer. And when we compare these two types of tissue, these are matched type of tissue, it means it's obtained from the same patient, okay? When we compare these tissues, prostate versus cancer versus benign, we found that GLUT5 was overexpressed, significantly overexpressed in this type of malignant tissue. We found the same thing with GLUT9, both fructose transporter, and we also find some tendency with GLUT11. So the question at the beginning was, well, which one of these transporters is more active in this tissues or cell? So in order to explore that, we moved to in vitro models, and we started to use cell lines. We took a benign cell line and two malignant cell lines. One of them is a low aggressive cell line of prostate cancer, and the other one is a metastatic high aggressive prostate cancer cell line. And what we found in this, contrary to what we found in tissue, in these cell lines, we found expression of GLUT1, glucose transporter. And we explained that by two reasons. One of them is most of these cells are grown in glucose glucose-based media, yeah, with high concentration. Actually, if you look at the glucose concentration in a regular media, it's around 10 millimolar, which is crazy. Okay, but when we analyze expression of GLUT5 in the benign cell, we couldn't find detect expression of GLUT5. However, in the both malignant cell lines, we found high expression of GLUT5, and concordant with what we found in tissue, also we found high expression of GLUT9. Okay, so in order to see if these transporters were actually, or some of these transporters were active, we analyzed uptake of glucose, radioactive glucose in these cell lines, and radioactive fructose. And concordant with the Western blot analysis, we found uptake of glucose in the 3 cell lines, which was very similar in all of them. But when we studied the uptake of fructose, the uptake of fructose was significantly higher in both cancer cell lines when compared to the benign cell line. Okay? So in order to explore from the kinetic point of view, from the functional point of view, which one of these transporters was the one that was mediated this fructose uptake in these cells, we did a kinetic analysis. We did a time course analysis and a Michaelis-Menten analysis, transport analysis, okay? And what we found here is that we detected a single component, which is determined by this graph, the Lindweber-Berg graph. We detected a single component, which is reflected here by a single slope, with a slope or Km around 6.8 in link-up cells and 7.1 millimolar in PC3 cell line, which is pretty close to GLUT5. So that means that in these cell lines, we have only a single component, a single functional component with the Km that is consistent with the expression of GLUT5. So even though we have expression of other transporters, the one that is mediating the fructose uptake in this cell line is GLUT5, okay? So then we started to analyze the biological role of fructose in these cell lines. And we performed a growth curve analysis in the presence of glucose, fructose, and we used galactose as a control, which is another exose. And what we found that in this analysis is that both cancer cell lines have the ability to grow either in the presence of glucose or in the presence of fructose. This is the only sugar present in the media. We use media without sugar, and we added 5 millimolar glucose or 5 millimolar fructose, okay? But that wasn't a generalized event for all exos, because when we use galactose, we couldn't see this potentiation of the proliferation. We observed the same thing when we analyzed K67 expression, which was practically the same. And when we analyzed migration and invasion, we found that fructose significantly promotes invasion in these cell lines when compared to glucose, okay? Which was something very interesting for us. So in order to prove this in a more in vivo study, we use a cell line xenograph model in which we use PC3 cell lines. And we provide this, we injected these cell lines in immunocompromised mice. And we feed these animals with glucose or fructose in the water compared to control condition. And what we found is that the fructose was able to promote the growth of these tumors when compared to control condition or even the glucose condition. Okay, so here we were seeing something different than we saw in the in vitro analysis, which was very interesting. And so to prove this in a more close model to what you're seeing in human, we use another type of xenograft model, which is the patient-derived xenograft model, in which you obtain samples from the prostate cancer patients, okay, and you transplanted them to immunocompromised mice. This is the PDX model. And what we found here was something consistent with what we found in the other xenograft model, is that in the presence of fructose in the animals fed with fructose in the water, in the drinking water, the proliferation of these tumors increased when compared to either glucose fed animal or control condition. Okay, and that was associated also with overexpression of GLUT5 and GLUT9, and not other transporter. Okay, so our conclusion is that of our study is that prostate cancer cells have the ability to take up fructose through GLUT5, and second is that fructose promote prostate cancer cell growth and aggressiveness in vitro and in vivo, and might represent an alternative energy source for prostate cancer cells. So what are the things that we're doing now? Well, this is a paper that is in third round of revision in cancer research, and some of the things that one of the last reviewer asked us is to provide some molecular mechanisms to unravel the role of fructose in these biological events. And one of the things that we are doing now is we are doing RNA-seq analysis and metabolomic analysis in order to prove this point, and hopefully we will have the results soon, and we were able to respond to the reviewers. So these are the current members of my lab: Carolina Echeverria, Emilia Flores, Carolina Gaete, Tina Meneses, and Vanessa Silva. We have some collaborators also, and we have collaborators around the world, most of them in the United States, and some of them also in Chile. Okay, and I think that's all I have to show to you today. Thank you.
Thank you very much, Dr. Godoy. It was a very interesting presentation. So we will move to the next presentation. Our exhibitor number 3 is Dr. Manuel Varas. He's a biochemist from the Pontificia Universidad Católica de Valparaíso. Okay. He has a PhD in biotechnology from Universidad Andrés Bello and Fundación Ciencia y Vida. Manuel did his postdoctoral research in the Karolinska Institute, and studying the role of calcium signaling in the acquisition of cancer stem cells properties. Manuel came back to Chile in 2015 to the Universidad Andrés Bello and started his own research in exosome biology. He currently works as a researcher at the Center for Cell Biology and Biomedicine at the Universidad San Sebastián. His research focuses on understanding how the activation of different cell signaling pathways can modulate the biogenesis and secretion of extracellular vesicles with pro-tumorigenic activity from chemoresistance or cancer stem cells, and how these extracellular vesicles could transfer these properties to recipient cells, transforming their phenotype. So please, Dr. Manuel Varas, can you start your presentation, please?
Thank you, Miss. Can you hear me?
Yes.
And the presentation is okay?
It's okay.
Perfect, thank you. So
Thank
you for the introduction, and also thank you to Andrew to invite me to present in this symposium. 3 years ago, also, when I'm back to Chile, or 4 years ago, I presented in a poster in ACCDiS. So for me, it's really important now to present my result in the last years. I want to start with this slide showing our campus. You can see for me, it's really beautiful, this campus. And it's in the middle of Providencia. So you can see a little microenvironment in the middle of a big city. And like this environment in Santiago, we can find a microenvironment in the tumor. And the tumor you can see here that is component by different type of cells. You can see here that, of course, the principal tumor, the main tumor here in green, but also you can see different type of cells, mesenchymal stem cells, fibroblasts, dendritic cells, natural killer cells, even extracellular matrix is a component of the tumor microenvironment. And one of these components in the last year that have been studied is extracellular vesicle, that in this slide you can see as an exosome. But what is extracellular vesicles? Last week, Andrew, Dr. Quest, and Dr. Lobos, they present their research, and they show what is exosome, but I want to show you again that exosomes or extracellular vesicles are a small structure that are secreted by the cells, and you have two ways to secrete these extracellular vesicles. One way is exosomes that come from the endosomal compartment. You can see here that, and the multivesicular body is fusioned with the cell membrane and release the content, and the content are the exosomes. Or also you can secrete microvesicles, or exosome also called, by the invagination of the cellular membrane. You can see here electron microscopy that you can see the multivesicular body with the interluminal vesicle, that when this is fusioned with the membrane, the exosomes are secreted, and also the generation of microvesicles. This one of the characteristics of these vesicles that are the size is between 50 and 150 nanometers in the exosome, and microvesicles can start in 50, but you can have bigger vesicles than around 1 micrometer as a apoptotic body. If we go to the cells, different stimuli can trigger extracellular vesicles biogenesis and release. For example, you can see here in this table that thermal stress, hypoxia, radiation, pH, or chemotherapy, some drug stimulation, gene overexpression can stimulate the biogenesis and secretion of extracellular vesicles. And in this context, several papers have shown that chemotherapy is one of these stimuli that trigger the secretion of extracellular vesicles. So our question is, or what we want to study, is the impact of chemotherapy in the biogenesis and release of extracellular vesicles. And this impact is responsible to the secretion of this type of extracellular vesicles called chemio-EDs. So to study that, we use two cellular models. First, we use this tumor spheroid, that, using this condition, this culture condition, and when we form this tumor spheroid, these are enriched in cancer stem cells. You can see here a flow cytometry measuring CD133, that is a marker of cancer stem cell, and you can see that this type of cells are enriching around 60%. And the other model is a cellular model that we transform a sensitive cells to a chemioresistant cells. We use this type of ovarian cancer cells at 2780, and you expose to a chemotherapeutic agent like cisplatin by 6 months with increased concentration of cisplatin, and this cell acquire chemoresistant. And you can see here the IC50 and I2780 is around 1 micromolar, but in the other cells, acquired chemoresistant is 12 micromolar. So what we want to study first is if these cells, the chemioresistant cells, have more multivesicular bodies or intraluminal vesicles that after that, when they are secreted, they are exosomes. So we will focus in this type of cells. And you can see here by immunofluorescence, when you compare the sensitive cells with the chemioresistant cells, these cells are not exposed to cisplatin. You can see here that the chemioresistant cells have more MBV-associated structure. And we evaluate that measuring CD63, that is a typical marker of exosome, and HRS, that is a protein responsible for the invagination of the interluminal vesicles into the multivesicular body. So it's important in the biogenesis of exosomes. And you can see here an increase of both proteins in the chemioresistant cells. But when you treat the cells with cisplatin by 72 hours, you can see here a decrease in both proteins. So long treatment with cisplatin decreased this MBV-associated structure in the chemioresistant cells. We treat with 1 micromolar, that is a low doses of cisplatin. We confirm that by Western blot, and you can see here an increase in HRS, relative protein levels in the chemioresistant cell, and the decrease when you treat with cisplatin. And we measure another member. This protein is a member of a score machinery, so it's a score 0 machinery. So after that, we measure TCS101, that is a member of the score 1 machinery. And also, we saw an increase in the relative protein levels in chemioresistant cells, and this decrease when we treat with cisplatin. So after that, we characterize the chemioVs, and for that, we use the 3 cells, the 3 model at 2780, 2780 resistant to cisplatin, and the chemioresistant cells in response to cisplatin. We treat these cells, we wait for 72 hours, and we harvest the supernatant, and also we did cell lysis, and we isolate the extracellular vesicles from the supernatant, and we did a Western blot and electron microscopy and nanotracking particle analysis, and also we did some Western blot in the cell lysis to see the enrichment of markers of exosome. So first, we did a Western blot, and we see typical markers enriched or typical proteins enriched in exosome, such as ALIX, TCG101, and CD63, and we compare against cell lysate, and you can see an enrichment of this marker, this protein, in extracellular vesicles in comparison with the cell lysate, and we use a negative control of GRP94, a protein that is not in exosome, and you can see here that is negative in extracellular vesicles and positive in cell lysate. We also did electron microscopy, and you can see here the typical morphology in the 3 conditions of the extracellular vesicles. And after that, when we measure the size distribution of extracellular vesicle in the 3 conditions, you can see here that is not really similar in the 3 conditions. It's around 160 nanometers. That is according with the extracellular vesicles society. Small extracellular vesicles are vesicles smaller than 200 nanometers. So we have our population enriched in small extracellular vesicles. We see the fraction of extracellular vesicles, and you can see here that around 70% is less than 200 nanometers, and around 30% is bigger than 200 nanometers. But when we measure the concentration of, or the release of extracellular vesicles in these cells, when we compare the sensitive or the chemioresistant cells, we don't see difference. But when we treat with cisplatin, we saw the increase in the release of extracellular vesicles in these cells. This effect is at 72 hours. So when we did the same experiment, but we recollect the supernatant and isolate the extracellular vesicles, but the treatment was only for 6 hours, this effect is not observed. And this is accompanied with the expression of HRS. Remember, when we have more secretion, we had less HRS protein at 72 hours. We saw a couple of slides ago. But in the case of when we treat only by 6 hours, the HRS protein level is maintained in the cell. So here we think that at 72 hours, the extracellular vesicles are secreted. They are secreted with this all component, TCFT101, HRS, etc. But when they are not secreted, the cells keep these proteins. We also did an electron microscopy of the cells, and we measured the interluminal vesicles into the multivesicular body. And at 6 hours of treatment with cisplatin, and you can see here an increase in the interluminal vesicles, so in the exosome, in the cells, chemioresistant cells treated with cisplatin in comparison with the other conditions. So if we summarize the result until here, we can see this. The chemioresistant cells have more ability to generate interluminal vesicles, so exosome, because they have more machinery to produce this, but they don't secrete more extracellular vesicles or exosome. But when you treat with cisplatin, this capacity to generate interluminal vesicles is higher, and also they secrete more extracellular vesicles. Why they secrete more extracellular vesicles? We think that it's a sum of the capacity of generate this, but also the chemioresistant cells have the capacity to generate a little bit more interluminal vesicles than the sensitive cells. But we believe that some proteins responsible for the exosome release are involved here. Only to show that we have this data comparing the sensitive cells and the chemioresistant cells, where we compare the expression of 3 different RAP proteins involved in the secretion of exosomes or extracellular vesicles, and we saw only an increase in the level of RAP27A in the chemioresistant cells in comparison with the other cells. So maybe in the chemioresistant cells in response to cisplatin, RAP27A increased more or is active in this condition. RAP22A and RAP75, they are not increased. So what is important now for us is see if the chemioexosome have some effect in the hallmark of cancer, like resistance to cell death on tumor microenvironment and invasion, influencing crosstalk between stromal and cancer cells. So we measure different protomorgenic properties of these extracellular vesicles, these chemio-EVs. So for that, this is the question, see if chemio-EVs could induce protomorgenic properties. So to study that, what we did was culture the sensitive cells first with the different extracellular vesicles derived from the different conditions. Here we have the chemioexo. And after that, 66 hours 16 hours after that, we treat with cisplatin the cells, and 48 hours after the culture with the extracellular vesicles, we measure cell death analysis. First, we did by trip and blue. We can see that the cells treated with extracellular with chemioexos protect the sensitive cells from the chemotherapy in comparison with the other condition. And we did the same experiment, but using by flow cytometry using light death staining, and we saw the same effect, that chemioexo protect the sensitive cells from the chemotherapy. In the same context, we did the same experiment, but 7 days after the treatment with the extracellular vesicles, we measure sphere formation. And in this case, we can see that chemioexo increase the sphere formation of the 827-80 cells. And this is important because these cells, the sphere cells, as I mentioned before, are enriched in cancer stem cells, and cancer stem cells have some chemioresistant properties. We also evaluate if these extracellular vesicles, the chemioexos or the chemio-EVs, can modulate another member of the tumor microenvironment like mesenchymal stem cells. So we culture this extracellular vesicle with bone marrow mesenchymal stem cell, and 48 hours after, we evaluate migration analysis by express assay. And you can see here that the chemioexo increase the migration of the mesenchymal stem cell in comparison with the other condition. And the migration of mesenchymal stem cell to the tumor have been related with the acquisition of protumorogenic properties. So we know that these chemio-EVs are protumorogenic. So of course, the cargo of these EVs are the responsible for these properties. So we want to study now which is the cargo of the chemio-EV. We did for that mass spec of the extracellular vesicles derived from the different groups. So we treat with cystatin or not. And after that, we recollect the subordinate and isolate the extracellular vesicle and did the mass spec. You can see here the protein, the number of protein that we identified in each group. And when we did gene ontology of this protein, we compare only the, here is the comparison between the extracellular vesicles derived from chemioresistant cells in black and chemioresistant cells in response to cisplatin in gray. And you can see different proteins related, for example, so chromatin silencing, regulation of gene silencing by microRNA, chromatin ensemble. Here you can see protein DNA complex, etc., single-strand RNA binding, transcription cultivator activity. But when we took this data and we did the interactomic analysis, we found this, that the protein in the chemioresistant cells in response to cisplatin have proteins involved in DNA double-strand break repair. So have some sense because this protein could help to the sensitive cells to acquire chemoresistance. And also some protein that regulate RANX2 expression activity, P10 stability and activity, or metabolism of RNA. All this stabilization of P53, specifically degrading P53. So all these characteristics are involved in the acquisition of protumorogenic properties. And in the middle of all these characteristics, all these protein, we have this protein that is UVA52 that we found that is important in the maintenance of stem cells. So maybe this protein is involved in the maintenance of cancer stem cells in our cells. So as a summary until here, we can see that as a chemioresistant cell have more score protein and another protein. Also we measure ALIX and is increased, and CD63, and also RAP27. But when we apply cisplatin to the cells, we increase the biogenesis of extracellular vesicles and also the secretion of these chemio-EVs. So which pathway could contribute? Because cisplatin maybe is activating some pathways, cellular pathways, and we know that mTOR mediates a mechanism of chemioresistance in ovarian cancer cells. So we want to see if mTOR is involved in all these effects produced by extracellular vesicles, by cisplatin in the ovarian cancer cell. So what we did was treat with rapamycin the chemioresistant cells first, and after that treat the cell with cisplatin, or rapamycin alone, or rapamycin and cisplatin in combination. And 72 hours after that, we recollect the supernatant and isolate the extracellular vesicles, only to show here that cisplatin activate mTOR in these cells, in the chemioresistant cells, and rapamycin, of course, inhibit the phosphorylation on this protein that is phosphorylated by mTOR, that is S6K. But when we measure extracellular vesicles, here we have effect of cisplatin in our cells that increase the secretion of extracellular vesicle, but when mTOR is blocked, this increase is blocked too. So until now, we know that mTOR contributes to the release of chemio-V, but also we did an experiment to see if the property of the chemio-Vs, the protomorogenic property of chemio-Vs, are also mediated by mTOR. So we did the same experiment. We took all extracellular vesicles from the different groups of the chemioresistant cells. And after that, we treat with cisplatin the sensitive cells. And after that, we did the same experiment. We measured trypan blue, live dead, and Nanexin-5 in the cells. And you can see here the protective effect of the chemioexo or the chemio-Vs, but this effect was blocked by rapamycin, measured by trypan blue, by live dead. And you can see here also that when the cells are treated with cisplatin, with the extracellular vesicle derived from cells treated with cisplatin, so the chemioexo, the cells are die less. Yes, you can see here because we have less Annexin-5. But when we block with rapamycin, this effect is blocked too. So indeed, to finalize the presentation, we can see the same. We increase all this protein, and cisplatin activate mTOR, and maybe mTOR, I don't know how, but we are studying that, are involved in the biogenesis of extracellular vesicles and secretion of this chemio-V with protomorogenic properties. The acknowledgement to all the group and the people to do the experiment: Albano, Felipe, Cristobal, Sergio, Organel Fagilab group, that is the Patricia Burgos lab, and Rodrigo Acuña from Universidad del Desarrollo, and of course the finance. Thank you very much.
Thank you very much.
Yep, thank you, Manuel. We will now hear the presentations related to microbiome in cancer. So let me introduce our new speaker, that is going to be Dr. Denise Bravo. She's a biochemist and a PhD in biochemistry from the University of Chile. She completed her postdoctoral training in the Laboratory of Cellular Communications in the Faculty of Medicine at the University of Chile. She's now an associate professor in the Department of Oral Pathology at the School of Dentistry, where she has been working since 2011. Dr. Bravo directs a line of research dedicated to the study of the pathogenicity mechanisms of oral and gastrointestinal bacteria. She has received distinctions, among which the Oriana Ayuso Award was awarded to her by the Faculty of Chemical and Pharmaceutical Sciences at the University of Chile. Among her academic positions, she has been the director of research of the School of Dentistry between 2013 and 2016. And she's the director of the research and development of the North American company UBIOM in 2017. So Denise will talk today about Porphyromonas and Helicobacter pylori in oral cancer. Thank you, Denise.
Thank you very much. Okay, we will move to the bacteria's world now. And let me to introduce you to Porphyromonas gingivalis, which is a gram-negative and anaerobic bacteria. This bacteria is able to internalize to gingival epithelial cells and possess several virulence factors like fimbria, capsule, gingipains, which are proteases, and the LPS, among many others. This bacteria has has a key role in periodontitis, and also a few years ago has been associated to oral cancer. This bacteria, P. gingivalis, can promote a process called dysbiosis and chronic inflammation in the subgingival biofilm. When we have a symbiotic microbiota in the subgingival biofilm, and P. gingivalis is introduced in this symbiotic microbiota, the community of bacteria start to change, start to decrease the commensal or the good bacteria, and started to grow or increase the amount of pathogenic bacteria. So the community start to change into an inflammophilic microbiota, and this activate the inflammatory signaling pathways and start to provoke inflammation, which is a characteristic of periodontitis. This process generate a tissue breakdown, which is used also as a nutrient for this inflammophilic microbiota, and the cycle can continue. Sorry. A few years ago, we demonstrated that the O antigen region of the LPS of P. gingivalis is required for inhibition of apoptosis in gingival epithelial cells. As a reminder, the outer membrane of the bacteria possesses this molecule, which is the LPS, and the LPS have several regions, and the most external one is the O antigen. So we have in the lab the wild type strain, which is this one, and also we have a mutant in the O antigen region. So we infected oral epithelial cells with both, with the wild type and with the mutant in the antigen region. And we observed with the wild type strain that the proliferation, the cell viability in oral epithelial cells increased and the apoptosis decreased, which is not observed when we infected with the mutant in the antigen region. We can see here. And then when we complemented the mutant and restored the O antigen region, we can observe the same effect as the wild type. Also, we observed that this process is probably mediated by the TLR4 because we observe an increase in mRNA levels. The last year, we published a review in which we show the several routes inside the epithelial cells that are activated by P. gingivalis. So we show here that several of these routes activated by this bacteria, by P. gingivalis, are related with processes associated to cell transformation, like apoptosis inhibition, EMT transition, increasing invasion and migration, metastasis, and increasing proliferation. But the question is that not all the people carry P. gingivalis has oral cancer. So there are other factors that are contributing to the cell transformation. And maybe other bacteria are modulating these responses inside the cell, or maybe are modulating the virulence of PET gingivalis. So we started to investigate about the H. pylori in the oral biofilm, and we showed that this bacteria, H. pylori, is present in the oral biofilm. It's present in saliva and supra and subgingival biofilm, and this bacteria could contribute to several oral diseases, oral bacterial transmission, and affect the virulence of several species. And as we know, H. pylori is considered a type 1 carcinogen by the International Agency for Research on Cancer. We also investigated that the patients with chronic periodontitis, as we can see here, has an increased amount of Helicobacter pylori in the periodontal pocket compared with periodontal health individuals. Also, in H. pylori positive patients, we can observe an increase in the Porphyromonas gingivalis amount. And also, the two markers of periodontitis are more severe when we have H. pylori positive patients. Sorry. So our aim was to evaluate whether the interaction between H. pylori and P. gingivalis modified the virulence of P. gingivalis and its ability to promote transformation of gingival epithelial cells. To do that, what we had first was to grow in a common media, Helicobacter pylori and Porphyromonas gingivalis. The idea is that these bacteria can grow together. Helicobacter pylori is a microaerophilic bacteria and P. gingivalis is an anaerobic bacteria. So it was not easy, but Christopher Soto, which is the PhD student in charge of the project, did that co-culture. After 24 hours of this co-culture, we used selective conditions for growth only P. gingivalis, and then we obtained this P. gingivalis to perform biofilm formation assays and also to infect oral epithelial cells, and made assays of bacterial internalization, migration, and qPCR.
Acá lo que se muestra es un...
Sorry. I don't know what happened. So, in the... Sorry, I don't know why it's talking my... Okay. We performed these experiments and we observed that the biofilm formation is increased when we had the co-culture P. gingivalis, as we can see here, and also when we infected oral epithelial cells with the co-culture bacteria, also we have an increase in this ability to internalize oral epithelial cells. So the co-culture is increased in bacterial virulence. Then, and as P. gingivalis is associated with the induction of inflammatory molecules in infected cells, we detected interleukin 8 and TNF in oral epithelial cells infected with the monoculture of P. gingivalis and the co-culture P. gingivalis, co-culture with H. pylori. So in both cases, we observed an increase in the cytokines when we infect with P. gingivalis, but when we infected with the co-culture P. gingivalis, we observed a more increase in this secretion. Both cytokines, interleukin 8 and TNF, are associated to epithelial cell migration. And in the case of TNF, also in the acquisition of metastatic characteristic. And as we observe this, then we assign the migration ability of infected oral cells. And again, we use the PG monoculture and PG co-culture with H. pylori, and we observe that we have an increase in cell migration when we infected with PG monoculture, but the migration increased even more when we infected the cells with the co-culture. Then we wanted to demonstrate that some proteins are increased, some virulence factor are increased. I don't know what happened with my presentation, sorry. And we observed that 3 proteins are increased, 2 hemagglutinins and 1 gingipain, which are associated to migration and pro-inflammatory cytokines. And as we observed that the gingipains were increased, we wanted to demonstrate if this process of the increase in migration is associated, are associated with, are mediated by the gingipains. So to do that, we assign experiment with an inhibitor of gingipains, which is the DLCK, and we demonstrated that with inhibitor, the effect in the increase in migration was decreased. In the last experiment I will show you today is that we did experiment with also with the mutant of the O antigen because it was demonstrated that the O antigen is very important for the activation of the gingipains, and as we demonstrated that the gingipains are important in this process, we did the experiment with the O antigen mutant to evaluate the role of this region in this process. So as we can see here, when we did experiment with the mutant in the O antigen, we did not observe the increase in cell migration that we observed with the monoculture of PG and the co-culture of PG. And also we observed the same amount of cell migration when we use the co-culture of PG with the inhibitor of the gingipain. So the conclusion is that we observe a synergistic interaction between H. pylori and P. gingivalis, in that H. pylori potentiated the virulence of P. gingivalis and its ability to promote migration of gingival epithelial cells, increased gingipain expressions, and contributing to the modification of gingival epithelial cells organization. We'd like to thank to the grants associated to this project, mainly to the ACCDiS, which is a very important support in the last years, and also to Christopher Soto, which is the PhD student in charge of this, Anilei Joar and Dr. Andrew Quest, which are my collaborators, and Daniela Salinas and Darna Venegas from my lab. So, thank you very much, and sorry about the technical problems in my presentation.
Okay, thank you, Denise. We will move on to our last speaker of this session in Cell Signaling 2, who is Manuel Valenzuela. Dr. Manuel Valenzuela is an engineer in molecular biotechnology from the University of Chile and a PhD in biomedical sciences from the same university. During his doctoral thesis, he studied the molecular pathogenesis mechanisms of infection by the Helicobacter pylori bacteria, and he completed 2 postdoctoral stays, one at the Catholic University of Louvain in Belgium, in the area of therapeutic applications in cancer, and another one in the University of Chile, focusing on molecular mechanisms Molecular mechanisms of cellular microbiology. In 2016, he joined as an academic at the Central University, where he's currently an associate professor. Among his projects, Dr. Manuel Valenzuela has focused on studying the regulation of the expression of the survivin oncogene, and he has trained some students. Dr. Valenzuela is a collaborator of the research at ACCDiS. He's an collaborating researcher at ACCDiS Center. Dr. Valenzuela will speak about Helicobacter pylori urease in the development of gastric cancer and extragastric pathologies. Thank you, Manuel.
Thank you. Good morning. Thank you for this invitation. I'm going to talk about the role of Helicobacter pylori urease in the development of gastric and extragastric pathology. This is a collaborative work between some researchers in this list, including Andrew Quest, Lisette, Lorena Lobos, Denise Bravo, and Esteban Palacio. I'm going to introduce Helicobacter pylori. It's a gram-negative bacteria that colonizes the stomach of 50% of the human world population, and is commonly transmitted by person to person by saliva, and is acquired during the childhood and persists in the stomach for the lifetime of the host without an antibiotic treatment. Normally it's asymptomatic infection, but in some cases produces severe gastric and duodenal pathologies, including atrophic gastritis, mild lymphoma, peptic ulcer, and adenocarcinoma. And for the reason, H. pylori is considered a carcinogen, since infection increases the risk for developing gastric cancer. Gastric cancer is a multifactorial disease when a combination between host susceptibility and H. pylori virulence and diet contribute to cancer development. And the intestinal type is the more predominant histological type of human gastric cancer. And this intestinal gastric cancer evolves as a cascade of well-established histological steps described by Correa in the 80s, and consists in several histological steps, starting with not atrophic gastritis, followed by atrophic gastritis, intestinal metaplasia, dysplasia, and finally gastric cancer. As you can see here, H. pylori influence all the steps during this cascade. Bacteria produce several virulence factors involved in the colonization and the origin of the pathogenesis associated with the infection, and produce several proteins involved in motility and chemotaxis like flagella, or some cytotoxic virulence factor like BAC8 produce a vacuolation of the gastric cells associated with peptic ulcer. Also, the bacteria produce several adhesives necessary for the binding of the bacteria to hot cells. And also, the bacteria produce a type 4 secretory system necessary for the injection of some effectors like Kg and peptidoglycan. That these two effectors are very important for the induction of the transcription of pro-inflammatory genes. And perhaps the most important virulent factor is the urease hydrolyzed urea, and allows bacteria to resist the acid challenge of the gastric acid of the stomach environment. Although mainly is cytoplasmic, a portion of the urease is found associated with the surface of the intact bacteria. As you can see here is an immunogold technique, and this is a electron microscopy photography, and you can see here the urease bound to the bacterial surface. And this urease is essential for the colonization, since isogenic mutants lacking the enzyme are unable to colonize the mouse stomach. And this activity hydrolyzes urea to produce ammonium and carbonate. And this reaction is a gold standard to detect H. pylori in gastric biopsies. Despite this relevance in the role of urease in gastric cancer cascade is still poorly understood beyond its catalytic activity. However, today the sulfate bond urease has been associated with some functions such as inflammatory response, angiogenesis, immune tolerance, protecting against asthma in a mouse model of infection, and also urease can modulate the expression of mucin in the gastric cells. In agreement with this observation, we have revealed a new role of urease induction of HIF-1 alpha transcription factor. This factor is very important or essential for the epoxy-induced increase in glycolysis and angiogenesis in tumor cell, as well as normal tissue. And in hypoxia, HIF-1 alpha is a heterodimer formed by the alpha and beta subunit. The beta subunit is constitutively expressed, but the alpha subunit is degraded in normoxia by proteasome. But in hypoxia, the protein, the alpha subunit is stabilized, translocated to the nucleus to bind the beta subunit and recognize his epoxy response element present in some promoter of gene associated with angiogenesis, erythropoiesis, glycolytic metabolism, and survival and proliferation. Of note, also HIF is not only physiological roles, HIF is also overexpressed in several types of tumors, and including gastric cancer. This is a result from our work, and as you can see here, the HIF-1 alpha is induced by H. pylori infection, is transient, but however, the target, the mRNA target of HIF signaling are sustained increasing in the time, and this response was associated with a non-canonical HIF activity because we observed by silencing HIF-1 alpha that this activation of the transcriptional factor was associated with a cell cycle arrest, and this is a non-canonical activity of the HIF-1 alpha transcriptional factor. Next, we wonder what was the virulence factor associated with this process, and we tested several isogenic mutants, and we found a mutant in Kg, oncogenic Kg protein. Here is a reported assay. You can see the mutant is not able to induce HIF-1 alpha. We check this possibility with making a new mutant in another strain, but the strain, the behavior is similar to the wild type strain. So the question is, what actually was the virulence factor associated with this induction of HIF-1 alpha? This is a SDS page and Kumasi blue staining of proteic extract of the both strain. And as you can see here, some difference. These bands were less abundant in the mutant, and this band corresponding, we analyzed this band by mass spectrometry, and the bands correspond to the two subunits of the urease, subunit urea A and urea B, and we confirmed this observation, this finding with urease reaction, assay activity. We measure the activity, and by immunoblotting, we detect less amount of urea B and less activity of the urease in the mutant CAG-A. Next, we construct a new mutant in another strain, and you can see here the clones didn't express any subunit urea B or urea A compared to the wild type strain, and we made the infection of the gastric cell, AGS cells, and as you can see here, the mutant in urea B, in urease, was not able to induce He1 alpha, unlike the parental strain. Also, we use a pharmacologic inhibitor of urease, hydroxamic acid, and we observed that this induction was independent on the enzymatic activity of the urease. We know that urease was shown previously to bind to toll-like receptor 2 to trigger inflammasome activation and to produce immune tolerance. And so we pre-treat the cell with an anti-toll-like receptor 2 blocking antibody. And as you can see here, the induction of He1 alpha was abolished in the presence of the blocking antibody. This finding was very important because toll-like receptor toxicity is very important during gastric cancer development, and normally is toll-like receptor 2 is overexpressed in gastric cancer. So some question remains unclear. For example, we need to know what is the subunit involved in this or binds toll-like receptor 2, and we need to know also what is the role of NKB and downstream effector of toll-like receptor 2 in this process or associated with pathogenesis of H. pylori. Also, in the recent evidence suggests that H. pylori infection is related with several pathologies or extragastric pathologies, including some neurodegenerative disorders, periodontitis, asthma, anemia, insulin resistance, among others, thus expanding the influence beyond of H. pylori beyond the gastric niche. One possibility is the remote action of other membrane vesicles secreted by the bacteria. These other membrane vesicles can cross the epithelium to get some distant organs, and this is by the peripheral circulation, and producing systemic extra effect. This vesicle, like exosome, Helicobacter pylori produced during the natural course of the infection other membrane vesicles. Here is an electron microscopy from a gastric biopsy. You can see here is the bacteria producing vesicles, other membrane vesicles. And other membrane vesicles are normally produced by gram-negative bacteria like Helicobacter, and they are spherical bilayer membrane vesicles on a size ranged from 20 up to 300 nanometer in diameter, and they are implicated in several bacterial functions like improvement of bacterial growth, biofilm formation, virulence transfer, acquisition of nutrient microbiome interaction, among others. And in terms of composition, normally this vesicle contain several virulence factors, nucleic acids, other membrane components, lipopolysaccharide, among others. Like a first approach, we isolate vesicles OMB from a reference strain. Here you can see the electron microscopy, the light scattering characterization, and the size was ranged from 100 nanometers to 300 nanometers. And this is a proteomic analysis of the proteins containing in the OMBs. And as you can see, there's a lot of protein cargo in the OMB compared to the proteins from the total bacteria. And this is a shotgun spectrometry analysis, mass spectrometry analysis, and the most abundant proteins in this OMB are the urease subunits, the chaperonin grow L, and catalase. And this is a summary of the several papers, and I can say the protein cargo of OMB in the case of H. pylori contain all the relevant virulence factors of the bacteria. Therefore, OMB are proposed as potential factors that can amplify the virulence of the bacteria. This is a very preliminary study, but we inject this fluorescent label vesicle into the a mouse model, and here is a bioluminescent of the accumulation of the vesicles, and it's possible to observe that these vesicles are accumulated in the gastrointestinal tract of the mouse, and also in the brain or heart of the animal. This is very particular accumulation of the vesicles from Helicobacter pylori, because if you compare this is with a similar experiment using a vesicle from E. coli, the E. coli vesicle accumulate into the liver and spleen in the same animal model. So OMB some in conclusion, emerging or relevant megavirulence factors which transport diverse virulence factor, including an abundant amount of urease. So this is a robust approach to study the systemic effect of this vesicle. So in conclusion, urease toll-like receptor 2 signaling might be key during H. pylori-associated gastric carcinogenesis and for extragastric disease development. And importantly, H. pylori are emerging as a relevant virulence factor, which transport diverse virulence factors, including an abundant amount of urease. I would like to say thanks to the team of collaborators in this project, and of course to Anit for the funding for the experimental approach. Thank you for your attention.
Thank you, Manuel. So we start now with the round of questions. We have lots of questions and not a lot of time. So, with whom should we start?
Maybe with you, Lisette. I have a lot of question marks for you.
Okay.
Okay. Beta-3 integrin is required for Taiwan-induced migration of cancer cells. What about interaction with other integrins? One question.
Yeah, well, Taiwan actually interacts with more integrin, not just alpha V beta 3. It interacts with alpha 5 beta 1, with alpha 5 beta 3, with alpha X beta 2, M beta 2, but in fibroblasts and monocytes. But there are no other interactions that have been described in cancer cells yet. We haven't published our data yet, it's in the process. But importantly, I think most aggressive melanoma cells are those that express alpha V beta 3. So I think that tells us something. Thanks for the question. Should we go one and one, like not all to the same speaker?
Okay.
Because we have lots of questions for everybody. Maybe to Alejandro. Andrew asked, is increased fructose uptake associated with enhanced metastatic potential of prostate cancer, like in cells like PC3?
Yeah, well, we measured the micromets in the tissues of the host animal. And what we found was an increase in the number of micromets and the area of micromets when we use fructose. The other observation that we have, that we are currently pursuing, is that we observe a more spindle-shaped morphology when we culture the cells in the presence of fructose. And we have measured some analysis to characterize expression of the EMT markers, and we found that some of them are promoted in this condition. So that's what we've seen so far.
Okay, thank you. Maybe a question for Manuel Varas from Vicente Torres. Some RAP proteins involved in EV biogenesis, such as RAP22A, are induced by hypoxia and HIF factors. Is it possible that the effects of cisplatin on EVs are mediated by HIF?
Thanks, Vicente, for the question. The answer is I don't know, but we didn't see the expression increasing the expression of RAP22 in the chemioresistant cell type. But not, we don't know if maybe cisplatin can activate HIP-1 alpha and this HIP-1 alpha increase the expression of RAP22. We know that RAP22 is responsible of secretion of extracellular vesicles in hypoxic conditions. So we believe that maybe cisplatin can activate hypoxia in our normoxic conditions, but we want to evaluate. So if you want to do the experiment, Vicente, I'm more than happy.
Okay. So Denise, from Lorena Orostica, is it common for both bacteria to coexist in the oral cavity in Chilean population? Have you evaluated the effect of H. pylori alone on oral epithelial cells behavior?
Yes, there are several studies that show that H. pylori can coexist in the oral cavity with other bacteria, which is in the plaque, so it's established in the community. It's not just in a transition between the mouth and the gut. There's no so many studies in Chilean population, but there is a group in Universidad de Concepción with Apolinaria Garcia that study some population that carry Helicobacter pylori in the oral cavity. Also, we have some preliminary results in a clinical study that we are doing and was stopped because of the pandemic. And we obtained some results, and we obtained Helicobacter pylori in the oral cavity of several patients that has Helicobacter pylori in the gastric cavity. So the answer is yes, is common, but in Chile we have to study much more.
To Manuel Valenzuela now, from Marcela Hermoso. Do Chilean population harbor TLR2 single nucleotide polymorphism associated to H. pylori infection and a stronger inflammatory response?
It's a good question. I have no in mind a research from Chile, but the international research say that yes. The answer is yes. There's some polymorphism associated with H. pylori infection, a higher susceptibility to develop gastric cancer. So it's interesting to perhaps to study that if the polymorphism are present in the population here in Chile. It's a very good question. I don't know. I have no reference from Chile, but it's international literature that say yes.
Lisette, I have another question for you. It's an interesting question, I think. Could the expression of Taiwan be associated with a hormonal action in breast cancer cells?
Actually, I have no idea. There is a study where they relate adipogenesis with thyroid-stimulated hormone receptor signaling, and those studies demonstrate that Taiwan and this receptor are inversely related. But in terms of, and there is a correlation also with the presence of Taiwan in fibroblasts that can differentiate to adipocytes when Taiwan is not present, actually. So there might be a relation with hormones, but this has been only seen with thyroid-stimulated hormone, but not with breast cancer related hormones. Let's move on to someone else, Denise.
Okay, for Alejandro Godoy, do you know if some enzymes of fructose glucose metabolism like aldolase are upregulated in prostate cancer?
Yes, we measure the expression of fructokinase, yeah? And we found that expression of this enzyme is increased in some of the samples. What we found even more interesting is that when we culture cells in the presence of fructose, the expression of these enzymes tends to go up. When cells uptake fructose, fructose is phosphorylated immediately by fructokinase. When the cells uptake glucose is immediately phosphorylated by glucokinase, okay, or exokinase. So the thing is, when you incubate the cells with fructose, fructokinase increase and exokinase decrease. And when you switch the sugar to glucose, then fructokinase decrease and exokinase increase. So the cells is very plastic in terms of their metabolic activity and the ability to regulate the uptake and metabolization of this substrate, at least in prostate cancer.
Okay, from Jorge Diaz to Manuel Varas, you show that in A2780 cis, some RAP are upregulated. What are the consequences of this RAP upregulation?
Thank you, Jorge. We think that the RAP27A that is upregulated in the chemoresistant cells maybe are not active. So cisplatin can activate this RAP and promote the secretion of extracellular vesicles. And if you can see in the when you saw the exosome secretion in comparison with the sensitive and with the chemoresistant, you don't see difference in the secretion of extracellular vesicles. Only when you put the cisplatin in the cell. So maybe the cisplatin is inducing the activation of the RAP. And I think you asked also for the RAP5 and RAP7. We don't know in the cell what happened with RAP5 and what with RAP7, but in the extracellular vesicles derived from the chemoresistant cells, not in response to cisplatin, we have the presence of RAP7 at RAP5, but not in the sensitive. So maybe it's overexpressed in these cells.
Thank you. From Marcela Hermoso to Denise, I wonder if there is any evidence showing the cytokine profile of oral mucosa infected by both pathogens. Is it known the infiltrate characteristic in different stages of the infective malignant sequence?
Yeah, it's not known yet. The study of Helicobacter pylori in the oral cavity is I think it's starting now. But it's a very interesting question that we have. Actually, the next step of this investigation is to study actually what happened with Helicobacter pylori virulence with the co-culture with PT. So we want to study the effect in the cells with Helicobacter pylori and pedingivalis, and to measure the profile of the cytokine is something that we want to do. So Marcela, if you are interested, you're welcome.
Well, we have a lot of questions. We won't be able to go through all of them. So let's see another one for Manuel Valenzuela. Do you know how these HP OMVs could be targeted specifically to these tissues in the in vivo model?
Sorry,
I guess it refers to the extragastric, no? This is a question from Maria Fernanda.
Sorry, I didn't hear. Do you know how this could target specifically to this tissue? Oh, very interesting, but the OMB presents several adenosines, so these adenosines perhaps are responsible for the target of this specific. But it's interesting. We can produce some mutant to test this idea, but really I don't know exactly what is the adenosine. But it's a good question. I think it's possible to do in the future.
I don't know what to do. We have lots of questions, but we have no more time because it's, this section was up to 10 o'clock. So
yes.
we can go through another round of questions.
1, 2.
One for all.
One more question?
One more round.
Okay. For Lisette, I have another question here. If cancer cells act similar to astrocytes in the pro-inflammatory environment, how we can connect a similar treatment for both cases? Can chemotherapy affect astrocytes?
Chemotherapy is normally used to affect proliferating cells, and in our brain, cells do not proliferate. But when there is an inflammatory process, astrocytes particularly, actually, there are people that describe that they can proliferate, although there is a controversy about it. And indeed, the drugs that are going to use for glioma or astrocytomas are typically a very different drug than the one that you all mentioned here, like cisplatin or whatever is called temozolomide. And it is used with radiation therapy and surgery. But I'm not sure we can actually use same drugs and expect similar results.
Okay.
Let's see, another question for Alejandro Godoy from Laila. Have you analyzed the effect of fructose in other cancer cells, like breast cancers or melanoma cells?
Yeah. Well, that's a very interesting question. And the answer is no, we haven't. And but there is a couple of other studies published in other type of cancer, like pancreatic cancer and intestinal cancer, in which they have observed that fructose has similar effect that we found in prostate cancer. So it seems like it's a more spread characteristic in terms of cancer in general, not only focusing on prostate cancer.
Okay, Manuel Varas, this is there are a lot of questions here in this paragraph from America Campos, but she would like to ask what happens with the RNA cargo of these chemio vesicles.
We don't know, but we believe, thank you, America, that we believe that the cargo of RNA will change in the chemioresistant cell in response to cisplatin, because one of the things is mTOR regulates biogenesis of long non-coding RNAs. And also we have a lot of protein involved in RNA metabolism. So for sure, we think that the content of RNA in the extracellular vesicles will change. So we are doing that experiment now.
Okay, great. From to Denise, how can one be aware of this biosis and how can it be treated?
This biosis, okay. This biosis is a process that you can prevent. And for treatment, you can try to restore the community. You have to intake some probiotics, prebiotics, and in the most extreme cases, you can do transplant of microbiotic to restore the complete community, because it's a complete community, very organized. So it's a process to restore this biotic event.
Okay, thank you. And for Manuel Valenzuela, do you know if these OMVs can change its composition or its effect on cells when obtained from bacteria at different times of growth curve?
No, I have no report about the question. No, I'm not, but OMBs contain several mRNAs can be translated by another bacteria. I don't know if bacteria can capture OMB from another strain, but this interesting question is really the word of OMB from H. pylori is still very unknown, and we need to answer several questions. I have no a report about the question, and really, no, I'm not sure.
Okay, we have a note here saying that if your questions were not addressed in the oral session, please respond it in the chat, because there are a lot of questions. And so we need to finish. I think the invited speaker is already waiting, so we'll be seeing the video, and then so we have a 5-minute break in the meantime, and then we have to continue with the session. Thank you very much to all the speakers.
Thank you.
Bye-bye.
Bye-bye, thank you.
Good morning. Can you allow me to share the screen? Can you see my screen now?
Yes, I can see your screen well. Okay. Hello, Ail. We're on a short break. I think everybody will be returning in a couple of minutes.
Sure, thank you so much.
There's Alejandro now. Okay.
Okay. Hey.
Hello, Ail.
Hey, Alejandro, how are you?
Fine, how are you?
Good, good.
I'm waiting for instructions.
How is the COVID-19 in Chile? Is it as bad as here or not?
Now it's under control, but we think that we are in the summertime here, so
yeah,
the season is favor us in this time of the year.
Okay.
So we expect a couple of months with a low rate of infection. How are you doing in Miami?
I'm doing okay. I mean, it's infection is still high in Miami, but it's not as bad as the north of the country.
Okay.
Yeah. Because here it's a bit more hot, you know, and humid, but still we have a lot of cases here.
I know.
I mean, the north, we are having 126,000 new cases every day. It's a joke, you know. 125,000.
In winter time now.
Yeah.
Which is good for the virus, but for us.
Yeah, I mean, we are expecting to get to 200,000 cases every day. That's
too much.
And you know how the politics is in Miami, in the US, not Miami. The politics.
Very active politics.
So shameful.
In the news every minute.
So shameful.
We have been following all the situations.
The circus. I have been following the circus around.
Okay, I think I'm waiting. I waited for someone tell me, let's go. So, but I think we're ready, or not?
Yeah, let's go.
Check. I cannot check here if all people is back already. But okay. We're going to start with a plenary lecture. For me, it's a pleasure to introduce Dr. Wael Rifai, who received his MD in 1986 in Cairo, followed by a PhD in cancer genetics, 1996, from University of Helsinki, Finland. Then he moved to the United States, to the University of Virginia in 2000, and then to Vanderbilt University in 2005. And now he is currently, since 2017, in Miami, where he's director of tumor biology program at Sylvester Comprehensive Cancer Center. Dr. Rifai has been elected as a fellow of the American Association of Advances in Science. He leads NCI funded research in upper gastrointestinal tumors. His research interests cover molecular biology, functional genomics, target therapy, particularly in gastric and esophageal tumors. He has more than 190 research articles in high-cited peer-reviewed journals, such as Journal of Clinical Oncology, quienes, Cancer Research, Oncogene, and Gastroenterology. So for me, it's a pleasure, since we have been collaborating for many years, to introduce my friend, Dr. Wael Rifai, for this plenary lecture. Well,
thank you, Alejandro. It's really a pleasure to be speaking at this venue. I'd like to thank, of course, Alejandro, Andrew, and Gary for the kind invitation. I wish we could do it in person and see the beautiful San Diego, but maybe a different time after we all get vaccinated. The talk that I'm going to give today, let me know if you're hearing me well, if not, I can raise my voice. But yes. I'm going to talk to you about TFF1 as a gatekeeper in gastric cancer. And really, I'm going to summarize some of the work that we have been doing over the past many years to document that TFF1 is very important for the protection against gastric cancer. Before I forget, all credit is to my lab members who are shown here, who have been actively involved in this research, and almost all of them have been with me for many years, moved from Virginia to Vanderbilt to now Miami. So it's a pleasure to have all of them. Just to give you a summary of what we have in the lab to open any collaboration. I already have collaboration with Alejandro, but I'm always open to more. We study genomic hotspots in tumorigenesis, focusing in 21q loss, 17q, and 20q amplicon, mainly aurora kinases, A and B. The antioxidant response in esophageal carcinogenesis, inflammation, diet, and cancer, and targeted therapy and molecular resistance, and also microRNAs for early detection and therapeutic follow-up. My talk today will focus on the 21q loss of TFF1 and touching on the inflammation pathways. I think I will go fast about this. You all know about gastric cancer. You are familiar with that. Majority of gastric cancer that we have worldwide are the sporadic gastric cancer, accounting for 90% of the tumors. And they can be classified as an intestinal type or diffuse type gastric cancer. And the predominant one is intestinal type, although we have seen over the past few years an increase, an uptick in the diffuse type gastric cancer. Other types of gastric cancer include lymphoma, primary gastric lymphoma, gastrointestinal stromal tumor, GIST, gastric carcinoid, and then the familial type, which is really about 10%, mainly due to loss of E-cadherin due to mutation. But there are some other also genetic factor, but the most predominant one for genetic or familial gastric cancer is E-cadherin loss. This just to show you here about what are the risk factors for gastric cancers. And definitely infection with H. pylori remains the #1 risk factor, established risk factor. It's classified as a carcinogen by the WHO. And that really relates to tumors in the antrum and the body. Whereas if you go a little bit up, like tumors in the cardia or gastroesophageal junction, the risk factors change and become reflux disease and obesity. And of course, the junction tumors are more related to reflux disease. It's still a very lethal cancer worldwide. About 750,000 die of gastric cancer every year. Overview of H. pylori. It's a gram-negative bacteria, infects the gastric mucosa, and first discovered by Drs. Warren and Marshall while he was at University of Virginia. I had the pleasure to interact with him back then. And then more than 50% of the world population, 3.5 billion, 4 billion, you name it, are infected with H. pylori. and it goes through a cascade leading from gastritis, atrophic gastritis, intestinal metaplasia, dysplasia, and cancer. And lately, of course, the SPEM has been introduced also as an intermediate stage of gastric cancer. And from H. pylori is the CAG pathogenicity island, is the one which is really carries oncogenicity related to infection with gastric cancer. Over the years, we have done a lot of genomic discoveries, whether it's epigenetics or genetics, RNA expression, RNA sequencing, array CGH, and bisulfite sequencing. But this is to show you some of our early work showing that hotspots for gastroesophageal cancers, the red areas are amplification, green areas are losses, are distributed among all chromosomes, denoting that it's a very complex disease. So many areas are hit, and that makes it even difficult to come up with targeted therapy. And surprisingly, the mutation rate is very low in gastric cancer, meaning is, other than the P53, which is mutated in about 50% of gastric cancer and tend to be in chromosomally unstable disease, ACCDiS mutation, as I mentioned, is 11%, mostly the familial type, and all other mutations are really some 8–10%, and then it drops very quickly to 2–3%, making it very difficult to come with targeted therapy. In fact, targeted therapy has not been working in gastric cancer for the most part of it. And classification of gastric cancer based in the molecular subtype is the chromosomally unstable gastric cancer, which is characterized by P53 mutation in about 50% of them. Actually, it tends to be higher here when you get chromosomally unstable gastric cancer. And then there's a KRAS activation, and KRAS activation, we have found that's not really the mutation or amplification, but activation of the network is a feature of chromosomally unstable even in absence of mutation or amplification. Other subtypes, Epstein-Barr virus and macrocystic unstable or genomically stable, and genomically stable tends to be more toward the diffuse type and familial type gastric cancer. Most of my talk will be related to this type, chromosomally unstable gastric cancer. So trefoil factor 1, which is the topic of this talk, is really a secreted protein, which is present in an area, chromosome 21q, which is often deleted or has an LOH or methylated in gastric cancer. It's a protective protein secreted by the superficial foveolar epithelial cells in the upper bits of gastric glands. The family includes TFF2 and TF3, but TF1 is the one that is really very highly specific to the stomach and is lost upon progression. And we have found early on that TFF1 silencing is through different mechanisms, either through the deletion, methylation, transcription, also repression through the COBRA, and mutations, which are extremely rare, about 1%. Most of the causes related to TFF1 silencing are deletion, methylation. And you can appreciate looking at this, seeing that in the normal epithelial cells, you have a lot of expression of TFF1, and with progression to cancer, it's completely lost. And that's true in about 70% of gastric cancer. And when you do the quantification, we used about 500 tumor tissue at different stages as compared to the normal. You can see that it really starts very early on from the intestinal metaplasia. So in gastritis, actually, you have an overexpression of TFF1. So when human tissue samples have gastritis only, there is overexpression because it's a protective factor. But when it gets to intestinal metaplasia, dysplasia, there's a progressive decrease in the level of expression. So the first evidence we have for a trefoil factor 1 as a tumor suppressor came from the TFF1 knockout mouse model. And the TFF1 wild type has a very normal glandular structure, of course, but the TFF1 knockout can have a progressive lesions from the hyperplasia compared this to, you can clearly see the hyperplasia occurring in the gastric glands, which also progress in this mouse model to low-grade dysplasia, high-grade dysplasia, and also progressive adenocarcinoma shown here, infiltrating into the submucosal layer. So that was the first evidence that showing that TFF1 is a real tumor suppressor gene that its loss lead to development of gastric cancer. So we started a study where we have mouse and human tissue samples, and the idea was to find transcriptional changes or networks that are relevant to both human and mouse. And the reason we did that we believe that if we can see the same changes, that means they are very important for carcinogenesis rather being a noise seen sometime in human, because mice are genotypically, you know, similar. So we had the mouse 50 cases and we have human 200 cases. We crossed and we did some transcription and microRNA sequencing to find what's overlapping in the middle here. And we focused really on the middle here, what's really common between these two. Of course, there are a lot that can be done, and it's not the topic of this talk, so I'm going to skip them for now. And what we found, what was really interesting, that regardless you are using or mouse or human, these are the overlapping pathways networks, and showing here the number of gene signatures and that are related to the pathways. So as you can appreciate here, you can see that among these were STAT3, NF-κB, and beta-catenin came consistently between mouse and human gastric cancer networks. So that gave us a reason to study these pathways. And of course, inflammation, everyone knows how important is inflammation for many cancers, including gastric cancer, but especially for gastric cancer due to the infection with Helicobacter pylori. And the concept of inflammation of cancer is not by any means new, has been from the 1863, the famous Dutch German pathologist Rodolph Virchow, that chronic inflammation can really promote gastric, not gastric, but cancer in general. And I will focus here on the crosstalk between NF-κB and STAT3. So, and the evidence that we see a lot of inflammation also in our mouse model, which mimics the human, you can see it here, that this is a case of gastric cancer in the mouse. And you can see this is a submucosal cancer glands, and you see accumulation and a lot of infiltration of immune cells in the tumor microenvironment. So there is really a lot of inflammation going on. And we have this TFF1 knockout mouse model. So what happened to the inflammation? So you can clearly see here that when you add the wild type mice here, when you add H. pylori, definitely you can increase the inflammation score. But the TFF1 knockout mouse model, compare the orange to the green, just by knocking out TFF1, you have increased inflammation, which can be further induced if you add on top H. pylori. So this is denoting that in the mouse model, you have high inflammation, and with H. pylori, you can move it even stronger. And you can see that the cytokines TNF-alpha and IL-1 beta are induced in response to H. pylori, and they are higher anyway in the TFF1 knockout mouse model. So the link between H. pylori, the TFF1 knockout, and inflammation became clear from this slide. And is TFF1 really playing a role here? So in mediating or promoting the inflammatory phenotype in gastric tumorigenesis in response to Helicobacter pylori. So this is just showing you an experiment where we infect with the cells with either G166 or 713 H. pylori strains. They are both GAG-A positive. And you can clearly see if you reconstitute TFF1 in the cells, that you can bring the cytokine significantly down, the TNF-alpha and IL-1 beta, as you can see here in red color compared to the blue. Once you put TFF1 in the cells, the response to H. pylori infection is significantly compromised in terms of cytokine expression. So this shows us that really TFF1 plays a role in suppressing the pro-inflammatory signaling in gastric epithelial cells. So the STAT3, which is going to be the first part of my talk, and STAT3 is known to be involved with and contributing to the inflammation process and immune response, especially immune evasion and preventing apoptosis, stimulating angiogenesis, which are all important steps in carcinogenesis. And it plays a very important role for survival and drug resistance. You can see a lot of literature about that STAT3 activation is not only related to survival, but immune evasion, but also resistant to several chemotherapeutics and targeted therapy have activation. And most of the times due to the canonical pathways where the IL-6 really binds to IL-6 receptor complex, which include the IL-6 receptor itself and GB130. And this leads to activation, dimerization of JAK, which will phosphorylate STAT3, and STAT3 goes to the nucleus. So this is the known pathway. I'm going to study this area here and show you some data. So to figure out if STAT3 is really important, as we see in our genomic discovery, we also did the mouse and human gene set enrichment analysis, and you can see clearly that the STAT3 signature is significantly enriched in the tumor set, whether you are in the mouse or in the human. And when you do the analysis of several of STAT3 target genes, which many of them here are shown, you can see that the knockout mouse model has activation or overexpression of several of these STAT3 target genes, and examples are shown here: VEGF, c-Myc, BERK5, and so on. But what's important that when you see this across different stages, whether it's dysplasia or non-dysplasia in the TFF1 knockout mouse model, you can see that in non-dysplastic versus dysplasia, that's clearly it gets higher as the tumors progress or as there is progression, indicating that this is really a progressive event happening in the TFF1 knockout mouse model. And when you do the human study, you can clearly see the inverse correlation between the TFF1 expression and STAT3. So normal stomach, you can see here, TFF1 is very high, STAT3, nuclear STAT3 is very low or doesn't exist, only very few cells in any given gland. But in cancer patients, in cancer sample, you can see TFF1 is lost, and in the same gland, you can see that there is accumulation of nuclear STAT3. So that's showing us that there is really a very nice inverse correlation also in the human. Is there a causal relationship that we can confirm between TFF1 and STAT3, and what does it do in response to IL-6? Because IL-6, of course, there are many other cytokines that can activate STAT3, but IL-6 is one of the most important ones, or at least the one that has been most studied. And you can see here, this is gastric cancer cell line, and then we did reconstitution of TFF1. And once you put TFF1, you see that STAT3 has gone out of the nucleus. You can clearly see that here you have significant reduction compared to here. That nuclear staining here is all non-nuclear staining. And in following stimulation with IL-6, you can see that you can get almost all cancer cells will have activation of STAT3 being in the nuclear. Whereas if you have TFF1 reconstitution, you really don't see any nuclear STAT3 or very little amount of nuclear STAT3. And that's giving us, and the quantification is showing here, that even with IL-6, you get the induction, but look at this with TFF1, there's almost no induction of immunonuclear STAT3. So now this is kind of a proof of concept that really TFF1 was critical in suppressing the STAT3 signal, and lots of STAT3 is also very important for activation of STAT3 in the gastric epithelium and gastric cancer. So we did here some organoid cultures, and we did the TFF1 knockout organoids, and you can see here, this is the structure of the organoids shown for you here, and this is just zooming in, showing you here that TFF1 has really a lot of STAT3 that is in the nucleus. Whereas if we have the conditioned media from cells expressing TFF1, you can see that you can completely remove, or there's no more STAT3, nuclear STAT3. Or if you add to this TFF1 knockout organoid, you just add the TFF1 recombinant protein. So you put the TFF1 recombinant protein in the cells, and you can clearly see that there is no more any nuclear. Compare this one to this one. So that's clearly also proof using the organoid models that TFF1 can inactivate STAT3. And it's important to check the reporter transcription activity. So that's showing you that STAT3 reporter confirming the same thing here, that with IL-6 you get induction of STAT3 transcription activity, whereas if you add TFF1, the transcription activity as measured by STAT3 luciferase is significantly down, and the expression of target genes is also induced by IL-6, but the blue indicates that it's significantly down if you reconstitute TFF1 or add a recombinant TFF1 on the cells. Long story short, I don't want to go into many details, but the long story short here is that we found that really TFF1 interferes with the formation of this complex here, the IL-6 receptor GB130 complex. And the way it does that it binds to the IL-6 receptor. So we did a proximity ligation assays to figure out what's going on. And we can clearly see that in response to IL-6, if you stimulate the cells, then GB130 and IL-6 receptor are binding to each other as denoted by the red color here. So this is a proximity ligation. So the system is working. If you put IL-6, you get GB130 binding to IL-6 receptor, and you get the activation of STAT3. Whereas if you put recombinant TFF1, you don't see any of this red signal. That means that IL-6 receptor is no longer binding to GB130. So now we know that IL-6 receptor is no longer binding to GB130 in response to IL-6 if you put TFF1. So how does this happen? So we did several proximity ligation and also co-immunoprecipitation assays, and we found that TFF1 can bind to the IL-6 receptor. So you can see here that in absence of TFF1, so of course everything is negative. TFF1 IL-6 receptor is negative because you don't have TFF1. But the minute you add TFF1 here, you lose the binding between GB130 and IL-6 receptor, and then you have binding between TFF1 and IL-6 receptor, which indicate that TFF1 binds to IL-6 receptor, and that by that shields the receptor from the formation of the IL-6 GB130 IL-6 receptor required for activation. So this is really was the mechanism here that TFF1 played in suppressing the STAT3 activation in gastric cancer. So summary one, that in vitro and in vivo studies are really important to denote that TFF1 is important for activation, for suppressing STAT3, and TFF1 interfere with the formation of the receptor complex. And we can conclude that loss of TFF1 is an important early step in promoting STAT3 activation, STAT3 mediated gastric tumorigenesis. Since our bioinformatic analysis also showed NF-κB as another important network activated in gastric cancer in both mouse and human, and we have the mouse model loss of TFF1, so we wanted to study if TFF1 can really also play a role in suppressing NF-κB signaling pathway. The canonical pathway, as you know, includes activation through TNF-alpha, which binds to TNF-alpha receptor, and then leads to phosphorylation of IKK alpha, that phosphorylates inactivates IκB, leading to the release, degradation of IκB, and release of P65 to go to the nucleus. It's a known pathway. But we are working with secreted protein, so we think it's about everything will be happening on the cell surface here. So everything will be happening at the receptor level because it's a secreted protein. And we also did the gene array, and we found that NF-κB is one of the most important transcription networks, also independent from the other study that I showed you. And we found that several targets of NF-κB pathways are activated in the low-grade, high-grade displays in the knockout. You can see many genes here, not just one or two. I'm showing many examples here, from TNF-alpha to IL-1 beta, CXL1, CXL2, and so on. Many of these are known, and they are also upregulated in the TFF1 knockout mouse model. So what we always like to do human tissue to see is this seen in human tissue, and we can see clearly here. This is very beautiful, one of my favorite slides actually. And the reason is when you have normal gastric glands, you have the TFF1 is expressed, but you go down here for the same glands, you don't see the expression of nuclear NF-κB. Whereas in the same core, when you have dysplasia, as shown here, you lost the TFF1 expression, and this gland immediately has nuclear NF-κB. So in the same core, you can see glands next to each other. You can see this inverse relationship in glands next to each other based on their histological subtype. And in cancer, of course, we lose the TFF1 and we get activation of NF-κB shown beautifully here. So there is really an inverse relationship between TFF1 and NF-κB. So we move into doing some ex vivo studies, and we did using the mouse tissue. So we did an ex vivo study, and they have a lot of nuclear NF-κB, as you can see here. And ZUA1 is just used to show you that these cells are epithelial. So you can clearly see these cells are epithelial and they have a lot of nuclear NF-κB. Whereas in the TFF1 wild type, you really don't have any nuclear NF-κB. That's another thing that I wanted to show you here. And then if you go in vitro and then you have a gastric cancer cell line like UGS or any others, doesn't matter to that here, but you can see a lot of nuclear NF-κB. You add the recombinant protein for TFF1, you lose this nuclear NF-κB, everything is blue. That means TFF1 can really suppress the nuclear localization of NF-κB. And in response to H. pylori, can also TFF1 protect against H. pylori the same way I showed you for stats. So in response to H. pylori 7.13 strain here, you see a lot of nuclear NF-κB. You add the TFF1, then you lose most of the nuclear. You lose significant number or significant amount of nuclear localization of NF-κB. And quantification is shown here. Compare using two different strains, G166 and 713 H. pylori strains. Compare the green to the blue, and you can see once you have TFF1 added on the cells, then you lose this nuclear localization. And this is just in vitro, also showing you the same thing, that's really what's happening here. Two strains of H. pylori, you add TFF1. This is actually, we did here adenoviral infection of the gastric cancer cells with TFF1. So you can see the TFF1 clearly here. You can see the CAG-A. This is the H. pylori in all these settings here. So you can see the control and adding TFF1, both of them infected with H. pylori. CAG-A is positive. And you can see that you lose phospho-NF-κB here if you have TFF1, but that's associated with loss of the signal of IKK alpha beta, as you can see here. So that means that really it's working through the pathway. And if we go up to the receptor, what we found is really TFF1 is binding to the TNF alpha receptor, preventing the formation of the complex with the TRAF. With TRAF2. So as you can see here that if you treat the cells with TNF alpha, then the TNF receptor is really binding to the TRAF2, which is here, which leads to the activation of the TNF receptor complex. You can see by co-immunoprecipitation, you can see the signal here. This is IB for the TNF receptor. You can see the TRAF signal. But if you add the TFF1 recombinant protein, it doesn't matter whether you activate or don't activate with TNF alpha. You don't get this binding between the TNF receptor and TRAF2. That indicates to us that also TFF1 is working on TNF receptor complex. So now I showed you two important pro-inflammatory signaling pathways, the TFF1, the STAT3 and the NF-κB that are suppressed by TFF1, and the way this works by the TFF1 binds to their receptor complexes and prevent the formation of the receptor complex. Have to note here that no one knows what's the receptor for TFF1. So is it really a secreted protein that binds to some receptors and some not? We don't know, but at least from the data we have so far, it can bind to two important receptors, the TNF receptor and the IL-6 receptor. And this really has never been shown before. If you recall, I showed you activation of beta-catenin in our network map. And beta-catenin, everyone knows beta-catenin is very important for gastric tumorigenesis. So not just gastric, for all GI cancers to that matter. And you can see in the TFF1 wild type, you can see clearly beta-catenin is where it should be. It's just localized to the membrane. And you can see a very nice staining for beta-catenin here. But when you have the TFF1 knockout mouse model, you get a lot of nuclear beta-catenin. It's very strong in nuclear beta-catenin, and this dysplastic lesion in the TFF1 knockout mouse model. And then you can appreciate here that also, of course, this just to show you that TFF1 is knockout, but you can clearly see that there is activation of c-MYC and AXIN2 and other genes related to or known to be transcription targets of beta-catenin. And we can do in vitro studies to try to investigate the mechanism. And what we found here that by adding, without TFF1, you have a lot of nuclear beta-catenin that you can see here in gastric cancer cells. And then when you add adenoviral infection with TF1 recombinant, with TF1 adenoviral particles, then you can see that beta-catenin becomes really in the membrane, and you lose most of the nuclei will be negative here, as you can tell. So that's showing us that, and then you can recapitulate this by doing the BFOB reporter or beta-catenin TCF reporter activity to measure the beta-catenin transcription activity, and you can appreciate that the black compared to the gray, that whenever you add TF1, there is significant reduction in beta-catenin activation. The mutant is used as a control, mutant beta-catenin, that's our control, but you can appreciate here that you can lose transcription activity of beta-catenin. And the same thing in response to H. pylori. So H. pylori can promote nuclear localization of beta-catenin. This has been shown by others and also by us. But you can clearly see that, yes, with H. pylori, you have a lot of nuclear beta-catenin. And then when you add recombinant TFF1 in the presence of H. pylori, beta-catenin shifts back from the nuclei to the cell membrane. Beautiful staining here of the cell membrane showing you that you lost the nuclear beta-catenin. We have nailed down the mechanisms related to BB2A, but this is just to show you that three very important signaling pathways, oncogenic pathways, are suppressed by trefoil factor 1. And this cartoon summarizes some of our work in this area, since I will be soon running out of time, that TFF1 acts on the receptors to suppress the TNF, alpha TNF receptor binding, and to suppress IL-6 receptor binding. So that's a secreted TF1, which can react to either autocrine or paracrine, IL-6 coming from either the same cell or neighboring cell or from the immune cells, and the same for TNF alpha, it doesn't matter, but it can prevent autocrine and paracrine activation through the canonical signaling pathways of this important signaling in response to H. pylori. And beta-catenin also showed you here, we don't know what binds, what's the receptor here, but we know that TFF1 plays a role in activation of BB2A to inhibit EKT and inhibit the beta-catenin. So we know that, but we still don't know what's the link between the BB2A and TFF1, what's really in the middle here, we have not figured out. We have done this, our work, and then we know in independent study that we have shown also that, I think this is also very important, I didn't have time to show, that TFF1 really plays a role in regulation of P53. So TFF1 promotes activation of P53. If you lose TFF1, you get induction of microRNA 504, and then you get induction of P53. We don't know what's the link between TF1 and microRNA. Is it an NF-κB or STAT3? Is microRNA 504 a target for NF-κB and STAT3? We don't know that. We have not investigated this that far. But we know that TFF1 plays a critical role in activation of P53. So I think I will bypass this, just showing you how to use the model for checking therapeutic response. I will skip all of this, and then I would like to summarize that gastric cancer cells, as I have shown you, contain multiple genetic and epigenetic alteration. I didn't focus on epigenetics here, but I showed you the transcription networks and the signaling networks in human and mouse gastric cancer. And this analysis of mouse and human is really very useful to understand the carcinogenesis. And a TF1 knockout mouse model has molecular and phenotypic similarity to human disease. It's important to note here that the presence of intact TF1 plays a critical role in shaping the chronic inflammation and tumorigenesis in gastric mucosa in response to H. pylori infection. So what we think is that TFF1, when there is the TF1 loss is a very important event, which is critical for mediating the oncogenic activity of H. pylori. And we know, I didn't show this here, that epigenetic regulation of TF1 is very common. And we know that DNA hypermethylation occurs as we age. And we know that gastric cancer also occurs as we age. Yes, infection with H. pylori can exist for 3-4 decades without gastric cancer. People diagnosed with gastric cancer in their late 50s and 60s. And by that time, there is more DNA methylation, and we think that's when the DNA methylation of TF1 hits, and they lose TF1, and that that really unleashes the oncogenic activity of H. pylori infection. Can we use TF1 expression as a predictor or a marker of an early event that be utilized for screening for gastric cancer and cancer risk? I think it might, we should, but again, I mean, I don't have access to enough samples to do this, or I don't have the power to do this, but just to show you that's really important. This is the members of my lab here that I showed in the beginning, some of my former members, collaboration with University of Miami and others, and of course, you know, I collaborate with Alejandro. He has been a very helpful colleague, and we have a couple of papers in high-profile journal, including Gastro Together for gastric cancer, and then a lot of collaboration in China, as you can imagine. I will be happy to take any questions. Yeah.
Great talk, thanks, Wael. Really appreciate it. There are some questions here in the chat. Let me see. 2 questions from Andrew Quest. Any idea if the other TFFs, there are, I think, 2 or 3 more, to similar effect to TFF1?
So the question is about, there are 2 more, TFF2 and TFF3. So TFF2 has been related mainly to spam, whereas TFF3 seems to be an oncogene, actually not a tumor suppressor. So ironically, TFF3 seems to be an oncogene and used to in colon cancer and esophageal cancer, and also some studies in gastric cancer. It's no longer a tumor suppressor. And TFF2 is related to the STEM phenotype. No one has studied any of these in relation. TF3 is out of the question, but no one has really shown if TF2 can do the same thing or not. And we didn't do it. So the answer, short answer, we don't know.
Okay. The question is TFF1 increase hicadin expression and thereby favor beta-catenin sequestration to the plasma membrane?
If you have TFF1, then beta-catenin will stay in the plasma membrane, yes. If you lose TFF1, there is now accumulation of beta-catenin. So the degradation complex, and we don't know exactly what happened. We know BB2A is involved in that. But we don't know what happened to the degradation complex of beta-catenin when you lose TFF1. Is it really that the TFF1 may be binding to this degradation complex and keeping beta-catenin at just levels in the membrane? I don't know. Yeah, as I mentioned, you know, the study of beta-catenin, we didn't dig enough deeply into it.
Okay. There's another question that is quite related, but I think it's interesting to know that two molecules from the same family have different actions. Like the question specifically is TFF3 acts as an oncogene, but TFF1 is a tumor suppressor gene. So how is that? Because they belong to the same family of proteins.
It's $1 million question. if I have an answer, yeah. I mean, and the thing is, if you think about, maybe if I can go back to the, I don't know if I can, let me try to go to, yes, you see this one, right? Can you see the slide?
Yes, yes.
So that's showing you that the expression in normal for TFF1, TFF2, and TFF3. So TFF1 is the red guy here. TFF2 is the yellow, and both of them are important for gastric tumors. TFF2 is more so for the SPEM. So the SPEM phenotype, the spasmolytic intestinal metaplasia phenotype, whereas the TFF1 seems to be important for the intestinal metaplasia. So TFF2 denotes a different type, which is the SPEM. But TFF3, as you can see here, it's not expressed in normal stomach at all. It's more so in the duodenum, small intestine, and large intestine. So that's why if it becomes overexpressed in gastric cancer, it doesn't act as a tumor suppressor anymore. It becomes acting as an oncogene. And I think it's really also maybe dependent on the level of the protein, because if you overexpress TFF1, you get different results. So TFF1 is known in the breast cancer, for example. There are conflicting reports in breast cancer and in ovarian cancer. When you have very high levels of TFF1, we think it's an oncogene. It becomes an oncogene. So it's just the just level or the physiological level of TFF1 which is a tumor suppressor. When you lose it, you lose this capacity. But forcing too much of TFF1 in other organ tissue actually leads to oncogenic activity and not tumor suppressor activity. So I think it has to be studied in a context-dependent and cell-dependent manner. So everything here is context-dependent and cell-dependent.
Perfect. Thank you. There's another question about is there any microRNAs that can affect TFF1 expression?
There are. We have not investigated them, but there are.
And I have a following question. Is there any germline mutation that can be associated with a clinical syndrome of TFF1?
No. To the best of my knowledge, there is no clinical syndromes related to TFF1 mutation. And TFF1 mutations, somatic mutation in cancer cells, occurs in about 1 to 2% of gastric cancer. It's extremely rare, even at the somatic level. Germline, nothing has been reported, or nothing that I have seen.
Okay, thank you. I have another question regarding the mouse model, because my understanding that is in general, mouse models don't reproduce the full gastric cancer cascade. They go up to dysplasia. In this case, your model is a full gastric cancer.
Yes. Yeah, but I have to, with a caveat here, that only about 10% of them will develop the invasive adenocarcinoma. So at the age of 6 months and beyond, only 10% will develop invasive adenocarcinoma. But you can boost this phenotype by two things. By H. pylori infection. So if you add H. pylori infection on top of the TFF1 knockout, then you can go from 10% to 20%. Will have the invasive adenocarcinoma. The other thing is high-fat diet. It's really that's what we are doing now. And high-fat diet can increase also, can double the incidence of invasive gastric cancer in this mouse model to 20–25% if the mice are having high-fat diet. So you can boost it, but at the baseline, it's only 10%. Low-grade dysplasia is all the mice, 90% penetrance, low-grade dysplasia or high-grade dysplasia, but the invasive cancer is only 10%.
Perfect. There's another question: if can exogenous TFFs rescue cancer progression? Is a future prophylactic treatment, for example?
That's something that actually there has been a group in Johns Hopkins, and remember 20 years ago, they were trying to do this clone it in carrots and some food that, the clone through genetic engineering of food products and trying to do this, but it didn't go well. As far as I know, they didn't succeed to get even the product itself. But can you use recombinant TF1? We tried to do this. We had a guy several years ago from Korea in my lab. He founded a company back in Seoul in Korea, and he's trying to do peptide delivery of TF1 and many others for the treatment of cancer using the HIV promoter to drive the expression. But nothing has moved forward also. So these are the only two attempts that I'm aware of that have tried to do this.
Perfect. Let me see if I have any other question. No, so far. I don't know if anyone has another question. Feel free to ask.
I'm happy to answer any question. I mean, you can email me anytime, and we can.
Here's another question. Is there any information that H. pylori chronic infection could induce promoter hypermethylation in
Yes.
TF1
Yes.
patients?
Yes.
Right. It's similar.
And that's why I'm thinking that when loss of TF1 occurs, that's when everything is unleashed. With aging is one, and also chronic infection of H. pylori leads to sustained activation of NF-κB. NF-κB actually drives the adenine methyltransferase 1, which leads to promoter hypermethylation of TF1 and many other genes. So, yes, short answer, yes.
So if you treat H. pylori infection, you can re-express the gene endogenously.
If you treat the H. pylori infection, depends, how far is this reversible or not. We know that there are a couple of papers showing that a lot of the DNA methylation becomes reversible after you eradicate H. pylori. There are papers 10 years ago showing that DNA methylation is reversible. But again, it depends if there has been LOH in one allele and DNA methylation of one allele, and then is this going to be reversible or not? But as a concept, yes. As a concept, yes.
Perfect.
And if the tumor, if you already have intestinal metaplasia and you eradicate H. pylori, then it's completely unpredictable whether this intestinal metaplasia will progress or not. And then they do excision biopsy and remove it, because that's what you need to do. But if you cannot see it and there are some hidden intestinal metaplasia somewhere, then they can progress. So depends when you eradicate the H. pylori and what was the histological lesion and what will be the endoscopic method of intervention.
Okay, here's another question on the same line about how H. pylori can cause methylation of TF1.
It's through activation of inflammation again. H. pylori by itself still induce NF-κB activation. Of course, loss of TFF1 will skyrocket this phenomenon. So if H. pylori can induce 30%, let's put it in a scale, for example, 30% inflammation score, then if you lose TF1, it becomes 80% or 90%. So there is still, H. pylori, even in the presence of TFF1, there is inflammation. And H. pylori in the essential stages, in the initial stages, there is induction of TFF1. See that? So if TFF1 is not genetically deleted and not methylated, so in response to H. pylori, there is actually activation of TFF1 induction. And believe it or not, TFF1 itself is a transcription target of NF-κB, meaning H. pylori induced NF-κB induced TFF1 to protect the inflammation. But if TF1 becomes lost due to methylation or genetic deletion, then NF-κB, this negative feedback gets broken. And then you get hyperactivation of NF-κB.
Perfect.
So actually there is a feedback loop between the three of them. And that's why TFF1 in most of the cases is really deleted or methylated. So NF-κB cannot induce TFF1, which actually acts as a negative regulator to put the brake on the activity of NF-κB, to put the brake on inflammation, because too much inflammation is not good for the cell.
Perfect.
But it gets complicated. I didn't want to go through all of this because it's...
No, I know.
It's really interesting story, yeah.
No, wonderful talk. I think there's no more questions. I think we have to move on.
Thank you so much for the invitation. Really, it's a great, and I see the beautiful. I don't know if this is Santiago or not behind you, but it's beautiful. It's beautiful Chile.
South of Chile. It's a wonderful place. So we should go there once.
Let's get the vaccine and go there.
Yes, absolutely. Really appreciate while your talk is a very illuminating.
My pleasure, Alejandro and Andrew. Okay, thank you. And Gary.
Thank you.
Thank you. Bye-bye.
Bye-bye. Thanks a lot.
Good morning, everyone. Let me introduce now the next session of our symposium, ACCDiS Symposium. This section is related to clinical research. Before introducing the speakers, I would like to introduce myself. I am Marcela Hermoso, and I want to just give a few comments to who join us now in this section about how it's going to work. The speakers in the session will be given his or her talk. There will be no questions, and at the end of each talk, I will introduce the next speaker. But there will be the questions at the end of this session, around 15 minutes or so. So as soon as we go through the talks, please write down your questions so you can also put it in the chat. So we have a really interesting session with speakers, Dr. Sebastián Moncada from the Universidad Católica with the talk of genetic classifier for the prediction of thyroid nodules, a biomarker developed model. Also, we have Dr. Hernan Gonzalez, also from Universidad Católica, with the study of targeting antisense mitochondrial non-coding RNAs for cancer therapy, lessons from preclinical trials, and also, and this is for Verónica Burcio, the last topic from Fundación Ciencias para la Vida.
Marcela?
Yes?
So I'll introduce you.
Yes, I will start with the talk.
Okay, let me introduce Marcela Hermoso, who is a PhD from Catholic University. She focused on immunology. She did a postdoc She did a postdoc in the United States and University of Nevada, and then an NIH. And in 2004, she joined University of Chile at the immunology program at biomedical science at the Faculty of Medicine. Currently, she's a full professor at Faculty of Medicine, and she mainly focuses on innate immunology, mucosal immunology, inflammation, and cancer, and she's part of the ACCDiS group in association with Dr. Andy West. Marcela?
Thank you very much, Alejandro, for your presentation, and I will share my talk, my presentation. So let me. This is so. Good morning again, everybody. So today I will share with you some studies on how alarmin IL-33 axis contribute to inflammation and cancer. As is known, the immune response mainly can be triggered by infection, as classically demonstrated, but also tissue damage and stress can activate immune response, particularly innate immune responses through apoptotic or necrotic cell death pathways, and particularly by pathogen patterns, moleculars, as damage-associated molecular patterns or DAMPs that can signal through TLR receptors or a related receptor named ST2 that can promote in physiological conditions to tissue repair and adaptations to stress. However, it has been shown in many models that these signaling pathways, when altered, can conduce to autoimmunity diseases or inflammatory processes. So these alarmins or danger-associated molecular patterns are endogenous molecules that are rapidly released following non-programmed cell death, but not by apoptotic cell death, and can, as I mentioned, in physiological conditions, recruit and activate receptor-expressing cells of the innate immune system and promote adaptive immunity. So the idea of these pathways is to restore homeostasis and wound healing and promote the tissue repair. Many molecules have been related to these functions as terms of alarmins, and as you see, many of these molecules shown in this table are endogenous molecules that are, when are released through necrotic cell death, can activate inflammation. And one of these molecules is IL-33, that is belonging to the superfamily of interleukin-1 cytokines. So IL-33 signals through a pathway through the receptor ST2. So it's an alarm, as I mentioned, activating several cells of the immune system, as you can see in this diagram. And IL-33 is released from structural tissues upon damage, mainly strong inflammation. And this alarming can signaling through IL-ST2 receptor, promoting, signaling pathway leading to growth factors, immunoregulatory genes, and mucosal healing. However, this receptor has been reported that through splicing, alternative splicing can generate a variant called named ST2S that can also bind ST2 IL-33, the ligand. So it has been shown in many chronic diseases that IL-33 and the soluble variant of this receptor ST2 is increased, like as initially in asthma, pulmonary fibrosis, coronary failure, trauma, and we demonstrated that in inflammatory bowel disease and other groups also, as well, and ulcerative colitis in the intestine, these molecules are increased, as well as in cancer. So IBD, inflammatory bowel disease, is a chronic inflammation that has a strong increase in prevalence worldwide, and is a multifactorial disease with several and heterogeneous clinical phenotypes. Unfortunately, it has no cure. Many patients have undergone surgery, so the quality of life of these patients are quite on risk. And the main interesting relation with cancer is that as it's a chronic inflammatory condition, as mentioned in the previous talk, there's a high association and a risk with colorectal cancer. In fact, patients undergoing many years of this recurrent inflammation in the colon and the ileum can have a increased cumulative probability of colorectal cancer, as is shown in this diagram, that after 10 years of diagnosis of the disease, the probability increased significantly. And inflammation is one of the triggers and one of the main causes of this colorectal cancer predisposition and probability of developing these states. So we wanted to see the relationship and the connection between these two diseases, IBD and colorectal cancer, and the role of IL-33-ST2 signaling pathway. Briefly, the IBD incidence, as I said, worldwide is increasing as in the last decades, as is shown in this map. But in Chile, there are no incidence data, but as you can see, this is a study of some centers in Santiago, clinical centers, that in the last 2 decades, the number of patients going to the clinic for and being diagnosed for these diseases, mainly ulcerative colitis, is rising from 2000. So it's an important issue to study in our country. So it has been shown that the ligand IL-33, the alarming of this signaling pathway, promotes intestinal homeostasis in animal models where are deficient of the receptor ST2, and it has been shown that the lack of its receptor, the colonic inflammation is more severe, and the Tregs regulatory cells are reduced in this model, together with an increase in the effector T cells. So also in patients, we and other groups who shown that is elevated in ulcerative colitis patients, mainly in the inflamed mucosa of the patients. So we wonder what is the mechanism regulating IL-33 in the intestinal inflammatory context. So one of the gene control, expression control are microRNAs that, as you know, can target several mRNAs and target genes and promote this degradation or block the translation. So some previous studies from other groups demonstrate that the microRNAs are deregulated in the mucosa of ulcerative colitis patients, and one microRNA calls strongly our attention is the microRNA 378A3P, because it has a seed sequence in the IL-33 mRNA. So our question was to see if there's an inhibitory effect of this microRNA on IL-33, and if the inflammation context of the disease can contribute to this regulation. So we characterize altered microRNAs and RNAs in ulcerative colitis patients' mucosa to see the role of the cytokines of these microRNAs, and we did this cross-sectional study using endoscopic patients with endoscopic score of 2 or 3, and also we compared with controls. And we found out doing microRNA arrays and mRNA arrays that the gene expression profiles were mostly modulated, those related to inflammation as TNF or gastrointestinal diseases. That is correct because we are studying this mucosa from these patients. And we then find out that in the microRNA array, the top 10 microRNAs were belonging to this family. These are all belonging to the microRNA that was downregulated in this previous study. So we analyzed that this the microRNA content in mucosa with in patients with this different Mayo score is reduced in those with inflammation, and Mayo 2 and 3 refers to strong inflammation, and these levels indirectly correlates with IL-33 RNA. And most mysteriously related to the inflammatory environment, we analyzed the levels of microRNAs and IL-33 in patients undergoing anti-TNF treatment. And as you can see, those comparing to those in inflammatory conditions and with another treatments, the levels of microRNA were reversed, increased, and IL-33 reduced. So there's a regulatory potential regulation of inflammation of the microRNA as we expected. So then we carry out some experiments in vitro using colonocytes and analyzing that the presence of it, if the microRNA can directly regulate IL-33 mRNA, and we did this analysis in vitro using a probe to see the binding of microRNA to IL-33 transcript, and we found out that, as in the control, this is negatively regulated. So it looked like the microRNA 378-3P could inhibit IL-33 expression. So going forward in some loss and gain of function studies in vitro using a cell line of colonocytes, we use a pre-MIR 33878 and the inhibitor, and we found out that the overexpression or inhibition of the microRNA regulate the IL-33 expression and cell viability, and also promote maybe healing process in the mucosa. So also we directly stimulate the cells with TNF-alpha, and we found out the microRNA is up-regulated and reversely regulate the mRNA of IL-33, suggesting these experiments that TNF lowers the levels of microRNA and promotes IL-33 production at the protein and messenger level. So in summary of this first part, we can see that microRNA 378P in inflamed tissue is decreased by TNF, that is the main cytokine present in the mucosa of these patients, boosting IL-33 expression, and this promoting this inflammatory environment. Also, as I mentioned previously, the soluble variant of the receptor of IL-33 is also increased in these patients, in the mucosa of these patients, as this is shown some years ago, that not only in the mucosa, but also in the serum, the levels of ST2 variant is increased, and these levels correlate with treatment and with the healing of the mucosa when such as the marker fetal calprotectin. So we suggest that this soluble variant of the receptor can be considered a marker of colonic inflammation, as well as reflects the severity of the disease, so can be considered a potential biomarker. So then we analyzed the levels of this variant according to the treatment of the patients, and for our surprise, we found out that patients receiving corticosteroid, the levels of this soluble variant is significantly increased in the serum, in particularly active patients that have a Mayo score of 2 or 3. So what is the mechanism regulating this soluble variant by steroid treatment or by excess of this steroid? Let me tell you that this gene is encoded by IL-1 RL1 gene, and has two promoters, a distal and a proximal promoter, that can regulate the production of these soluble variants. And in vitro, we found out that corticoids specifically regulate the soluble variant. This was done in mast cells that can express both membrane anchor receptor at the soluble variant, and it looked like these anti-inflammatory steroids specifically induce this soluble variant of ST2. So looking in more detail, we found out that this regulation is at the transcription level. As doing CHIP assays, we found out in these mast cells that the glucocorticoid receptors is enriched in the distal promoter in one region called GRE that binds and enrich the glucocorticoid receptor upon dexamethasone treatment. And as you see, this activity is upregulated by dexamethasone, that is a synthetic glucocorticoid. So this GRE that the GRE3 harbors a SNP that in the distal promoter, and we found out that the ST2 levels in ulcerative colitis patients are increasing, those harboring this risk phenotype, and the effect of the treatment can boost the production of this these variants, as well as in patients, as we see in vitro. And when we expose peripheral blood mononuclear cells to dexamethasone of patients harboring the different genotypes, we saw that a specific regulation of the soluble variant of ST2. So in summary 2, this, the ST2 gene can be transcriptional regulated at the promoter level, and genetic susceptibility can contribute to a deficient mucosal healing, and together with the steroid treatment. So finally, as I mentioned, that the IL-33 can be modulated in inflammatory conditions. We are developing an immunotherapy to balance the activity of this inflammatory pathways in the mucosa, and maybe projected to another inflammatory conditions. For example, in colorectal cancer, that we found that this axis is altered, as you see in patients having lymph node metastasis. The IL-33 is increased in patients with lymph node metastasis, and also associate the levels of in the mucosa, in the epithelial mucosa, that IL-33 associate strongly with desmoplasia. That is a more characteristic of colorectal cancer patients. And it was shown also in this study that we found that the presence of IL-33 ST2 components in the lymph node metastasis correlate directly with an unfavorable tumor microenvironment. That means an increase of tumor-associated macrophages characterized by the surface expression of CD163. So in summary, for this last part, IL-33 ST2 axis is implicated as well in colorectal cancer, as was shown, but we found out that this axis is mainly associated with desmoplasia, tumor budding, and lymph node metastasis, so can be an interesting marker and prognostic of for colorectal cancer patients. So let me thank the innate immunity people that are, most of them, Karen and Glauben, participate, as well as Marjorie de la Fuente, that she is an independent researcher now on clinical ACCDiS. Also David Diaz, that is now doing a postdoc abroad. And all our collaborators at national, collaborator from clinical ACCDiS, the Faculty of Medicine, Andrew Quest, Lisa Layton, and also from the immunology program. And thank you for the patients participating in this study. So thank you for the attention. And sorry. So now we move to the next speaker, that is Sebastián Mondaca. I want to introduce that his talk I made is entitled Pragmatic Trials in the Precision Medicine Era, Clinical Trial List Perspective. Dr. Mondaca got his degree in the Universidad de Valparaíso in Chile, and he got the specialty of medical oncology and internal medicine in the Universidad Católica de Chile. Afterwards, Dr. Sebastián Mondaca have clinical research course in Harvard University. And he completed as well in 2019, quite recently, a 2-year advanced oncology fellowship in Memorial Sloan Kettering Cancer Center, focusing on clinical research. And currently now is in the Faculty of Medical Oncologist and a physician scientist at the Pontificia Universidad Católica. Mainly his primary interest is in gastrointestinal and hepatobiliary cancer, and is dedicated in finding novel target and immune modulators therapy in gastrointestinal malignancies. His research is now focused on RB2, altered biliary tract cancer, and also is participating in the liquid biopsy program study plasma cell-free DNA, characterizing potential actionable targets in biliary tract. So please, Professor Mundaka, welcome and thank you for your participation in this symposium. And,
thank you very much for the kind introduction and for the invitation to present this interesting topic for me. So the topic is pragmatic trials in the precision medicine era. And first, I would like to start with my conflict of interest. Okay, so these are the questions I would like to answer during this 20-minute lecture. How did we get here in terms of the development of precision medicine? How should or do we evaluate the efficacy of precision medicine? And then some examples. I have been involved in biomarker-driven research in the academic setting, focusing on APC alterations in colorectal cancer and RB2 amplification in biliary tract tumors. Let's start. First, it's relevant to define what precision medicine is and what it is not. It is the use of biomarker to inform treatment selection, and this includes both a standard of care therapies and investigational therapies. It is not a test. Next generation sequencing has been very relevant for the development of precision medicine, but they are not synonyms. And it's not an approach for off-label drug prescription. Actually, most researchers are against to that practice. As I said, next generation sequencing has been very relevant. And here I would like to show the flow of the next generation sequencing program at Memorial Sloan Kettering. It starts with the patient consent, of course, so we can develop all the research aspect of this type of program. Then they collect both tumor and blood. They use the blood for the variant calling. They can filter the alterations if they also have germline material, but many platforms only use tissue. Then they prepare the library, continue with the sequencing process, and finally the bioinformatic analysis, which is key and it's very relevant for the correct variant calling. And you can see some difference between different platforms in terms of the sensitivity and specificity because of the bioinformatic analysis. And then you get a report. I'm going to show you an example here of the MSK impact report, which is the platform that they use at MSK. First, you have a summary on the top that briefly describe all the significant alterations that were found. Then the genomic alterations, they further characterize them if they are mutations, indels, copy number alterations, or rearrangements. And below that, the actionability, and they use a system I'm going to further describe in a few slides. And then the technical aspect for us as researchers and clinicians, it's very important, the coverage, how deep the sequencing was. And they normally report the median depth of sequencing, and also if there was a specific gene or segment of the gene that was below certain threshold of the depth of sequencing, because that's important for the sensitivity to find this type of alterations. Here I am showing a few examples of clinically relevant molecular alterations. Of course, we have mutations, indels, copy number alterations, and rearrangement. And all of these type of alterations are already a standard of care in terms of the analysis. And of course, because you can prescribe certain drugs to target that alteration in specific tumors. Here I'm showing a system to classify the actionability of genomic alteration. This was developed, was led by an MSK investigator. And here you can see that there are level 1 alterations. That means that both there is a companion diagnostic to detect the alteration that has been approved by the FDA, and also, of course, the therapy, the target therapy to drug that alteration. Then you have the 2A, in which you have a standard of care and FDA-approved target therapy, but there is no companion test. That means that there is no specific platform to detect the alteration that has been validated or approved by the FDA. And then you have mostly investigational alterations that you consider both how direct the evidence of clinical efficacy is, for example, if it is in the same tumor or if it is in another type of tumor, and how strong is the evidence, if it is a large clinical trials or a small clinical trials, or maybe some preclinical evidence. And that's how you can classify the actionability of the alterations. Of course, the actionability of the alteration is tumor specific. For example, one alteration can be level 1, for example, for breast cancer, but level 3A for another type of tumor. And here's how we would like this to work. You have a group of patients with the same type of tumor, and then you continue with molecular profiling, and then you can identify prognostic markers, and predictive biomarker, most important, that predict a response to certain treatments and to toxicity. So then you can group a subset of patients to receive a specific therapy. So this is the dream, the promise, however, not yet the reality. And I'm going to show you a little bit about the difficulties that there are to validate this type of strategies. So how should we evaluate the efficacy of precision medicine? One option is to evaluate the whole strategy. What is the real clinical utility of next generation sequencing, of doing this type of test? And you could run clinical trials considering 1,000 patients, and then they would undergo next generation sequencing, and a subset of those patients would have targetable alterations, and then you can randomize to receive a target therapy based on the NGS result or a standard of care therapy. That would be one option. And actually this trial has been done. This is the Shiva trial, a phase 2 trial that was published in 2015. It was conducted by a prestigious French group, and they considered many types of tumors, and they did a sequencing of those tumors and randomized to receive based on the alterations to a specific targeted therapy or a standard of care chemotherapy. And here you can see the Kaplan-Meier curves for progression-free survival, and you can see that there is no benefit with a hazard ratio of 0.88. There is no benefit of the targeted approach compared to the standard of care option. And what happened? How can we justify this type of results? Yeah, let's focus on, for example, this alteration, the mutations of PIK3CA, which were one of the most prevalent alterations that were found in this Shiva trial. Here I have these bars that represent different type of tumors, and they go from the tumor for endometrial cancer, which this type of alteration is around 40%, and then you go with a different type of tumors in which the prevalence of this alteration is less frequent. Here I'm showing a few specific PIK3CA inhibitors. There are many, as you can see, but none of these were used in the Shiva trial. They used everolimus for the patients who had this type of alterations. Everolimus is an mTOR inhibitor, and while mTOR is downstream PIK3CA, so there is rationale to use this type of approach, it's not a specific inhibitor. So this type of drugs did not exist at the time that this trial was conducted. So if you pursue this type of strategy, you would need to repeat the trials every 2 or 3 years as you are having more novel, hopefully more active therapies. So that's what a drawback from pursuing this type of strategy. Another option would be to do a large randomized clinical trials against a standard of care. And here I'm showing a study published in New England Journal of Medicine of maintenance olaparib. Olaparib is a PARP inhibitor for germline BRCA mutated metastatic pancreatic cancer. And let's see how many patients you'd need to assess to conduct this type of trials. In this study, 3,315 patients were assessed for eligibility, and less than 10%, 247, had this germline BRCA mutation. Of course, then the patients need to fulfill all the rest of the inclusion criteria. So at the end, you have 154, and those patients were randomized to receive olaparib or to continue with regular monitoring, which is a strategy that you can pursue after chemotherapy in pancreatic cancer. So here we can see the Kaplan-Meier curves of progression-free survival. There is some benefit, and this was statistically significant with a hazard ratio of 0.53. However, there was no benefit in overall survival. So let's see how many patients we assessed to recruit 150 patients to conduct this type of trials. It's of course a major effort. So it's of course an option, but it has many challenges. And the third option is to conduct pragmatic clinical trials, and I'm going to further discuss these options. One option are basket trials. In this type of trials, you consider multiple disease, different type of tumors, but these tumors have something in common. They have a specific genomic alteration, and of course, you would need some rationale that a target therapy can have a similar efficacy for this target alteration regardless of the histology. So then you treat those patients in a single arm trial and assess the efficacy using outcomes like response rate, duration of response, progression-free survival. Another type of pragmatic trial is the umbrella trials. In this case, you would have a single disease, for example, lung cancer. You perform genomic sequencing of these patients, and then you're going to identify different alterations, and you can divide the patients based on these alterations that you found, and every subset of patients receive a specific treatment based on the result of the genomic sequencing. So you have many single-arm trials that are based on the findings of the genomic sequencing. But of course, there are many challenges also when you conduct this type of trials. First, there are biological challenges. You need to establish that the mechanism of action is histology independent. And for example, BRAF V600E, it's a common alteration in melanoma. And there are specific BRAF inhibitors that are very active in melanoma. However, this same alteration is not as druggable in colorectal cancer, and this is because in colorectal cancer, when you inhibit BRAF, that leads to a negative feedback that reactivates the pathway through EGFR. That doesn't happen in melanoma, so the context does matter. So you need some evidence that the target therapy is going to work similarly across different type of tumors. And of course, like it's very challenging to recruit many patients with these low frequency alterations. You need to be very mindful about the clinical safety because you're not randomizing and not comparing with a standard arm, with a control arm. There are also statistical challenges because sometimes you can have different type of patients with different type of tumors, and some groups are going to be larger than others. And sometimes you can conclude that treatment is active based on the larger group, and because in some groups there are not many patients, sometimes you can conclude that the drug is overall active, including that subset of patients, but maybe the drug is not active in this small group, and you are using the good results in the larger group to justify the indications in a small group, which the drug is not active. So there are challenges there as well. And of course, there have been regulatory challenges because all the regulatory agencies have made efforts to adapt to consider these trials, because sometimes some drugs are so active that patients would not, and maybe it wouldn't be ethical to randomize when you have such data showing efficacy. It's hard to randomize in phase 3 trials. So some drugs that have demonstrated real clinical efficacy have been approved before doing a randomized trial, and I'm going to show you an example of that. NTRK fusions are a very interesting target. They are very, very infrequent, but in specific infrequent tumors, they are very prevalent. For example, in some pediatric sarcomas like infantile fibrosarcoma, most of these tumors have this type of alterations. And interestingly, they are very, very infrequent in very frequent tumors, for example, lung cancer or breast cancer. But in this type of tumor, less than 1% they have this type of fusion. NTRK is a tyrosine receptor, tyrosine kinase receptor. It's a neurotrophin receptor that is very important for neuronal development. This kind of fusions, they can lead to a constitutive active pathway that leads to angiogenesis, proliferation, and cell survival. So they can act like an oncogene. And we have this trial of efficacy of larotrectinib in TRK fusion positive cancer. And this is a small trial with only 55 patients. And as you can see, we have different type of tumors. Some of them are frequent tumors like breast cancer, but we have these other very infrequent tumors like infantile fibrosarcoma. And all these patients have in common that they have these NTRK fusions. Here we can see the waterfall plot. They represent the activity, the response rate of the drug. Here we have 0, and the bars that go down, they represent a percentage of response rate. For example, here we have many patients who had 100% of response rate of tumor shrinkage, and the bars that go up means a tumor growth. So as you can see, and the different colors represent different types of tumors. And so you can see that the drug was very, very active, independent of the type of tumors, as most of the patients had tumor shrinkage and many of the patients had complete response. So finally, I would like to discuss a little bit about biomarker-driven research in the academic setting. And first, I'm going to discuss this trial I was involved of APC alterations in colorectal cancer. As I explained, next generation sequencing can inform us about prognostic biomarking. And that is important because we can see here in this Kaplan-Meier curve how the prognosis of metastatic colorectal cancer patients has improved since the 90s compared to 2010. There's a very, very important difference. However, there's still a lot of variability, and even today we can see that some patients do die during the first year of treatment. So it's good to have prognostic markers so you can adapt and tailor your treatment. And so what we did is that we considered all the unresectable metastatic colorectal cancer patients treated at MSK between 2014 and 2017. We used this NGS platform to characterize the genomic landscape of these tumors. We used facets to assess tumor purity and loss of heterozygosity, and OncoKB as the classification I showed you to characterize the alterations. So from over 1,600 patients, we exclude all the patients who had resectable disease. So we end up with 471 patients that receive palliative first-line chemotherapy. These patients were grouped based on the progression-free survival they had. Patients who had less than 6 months, between 6 and 12 months, or more than 12 months. And we see how these genomic alterations distribute in these 3 groups. And we found that 2 genes were clearly different in one group from the other. For example, APC mutations were less frequent in patients who had a progression-free survival than less than 6 months, and more prevalent in patients who had progression-free survival over than 12 months. And the opposite happened with BRAF. So that means that APC, when it's absent, when there is no alteration in APC, patients tend to do worse. And this was also found in the multivariate analysis. We adjusted for some clinical features and other genomic alterations, and we can see that APC is protective, the mutation is protective, both for progression-free survival and overall survival. So the hypothesis was that APC is a very big protein. It's a tumor suppressor gene, and there can, depending on the location, the consequences can vary. And there is previous work showing that mutations before the 1400 amino acid leads to a non-functional protein, and that leads to high Wnt pathway activation, while alterations that occur after the amino acid for 1400 only partially inactivate the protein, leading to moderate Wnt pathway alteration. So we decided to explore how this translates into clinical outcomes. So here we can see the Kaplan-Meier curves that patients who had no APC alterations did worse. Then the patients who had late genomic alterations, and finally the patients who did better were the patients who had early APC alterations. So here we can see the concurrent alterations of these 2 groups of these early and late APC mutations, and we can see that mitogenic alterations, for example, KRAS, BRAF, and PIK3CA, were much more prevalent in patients with late alterations, 72 versus 46%, 8 versus 1, and 28 versus 11%. So what we are proposing is that probably late APC alterations leads to a lower degree of Wnt pathway alteration. So the tumor needs more mitogenic alterations that lead so the tumor can develop and become a clinically active tumor. So that would explain the different concurrent alterations between 2 groups. But even if we adjust for this type of alterations that have been associated with worse prognosis, this type of APC alterations still do worse than the early alterations. So probably there is a biologic phenomenon associated with these late alterations that is even more important than the concurrent alterations that they are associated with. And the second study I would like to discuss with you is related to the molecular landscape of biliary tract tumors. Historically, biliary tract tumors consider gallbladder cancer, intrahepatic cholangiocarcinoma, and extrahepatic cholangiocarcinoma. And this type of tumors, while they are different clinically, they have been lumped so you can have more patients and conduct clinical trials. However, the molecular alterations in these types of tumors are different. And here we can see that ERBB2 amplification is particularly frequent in gallbladder cancer. And since gallbladder cancer is very frequent here in Chile, when I was abroad, I decided to focus on this type of alterations. So what we did is that we explored all the patients who underwent molecular sequencing, all the biliary tract cancer patients, using this same platform of NGS. We classified genes as amplified if they had a fold change of more than 2. And there is a previous work of this institution showing a good correlation, concordance between NGS and immunohistochemistry for ERBB2 or HER2. So we analyzed 517 patients. Most of them had intrahepatic cholangiocarcinoma. And we can see that overall we found 14 patients with ERBB2 alterations, which were more frequent in gallbladder cancer as expected and less frequent in intrahepatic cholangiocarcinoma. Here I'm showing the lollipop plot describing the type of mutations that were found in this cohort. As we can see, the most frequent one was this S310F or Y alteration, which is an extracellular domain mutation. And actually this mutation, patients with this type of mutation has been reported to be responsive to certain treatments such as neratinib. And there are cases reported with breast cancer and this type of alteration, and even a few cases of biliary tract tumors. So it's interesting to know that this type of alteration is the more frequent one that we found in this cohort. And here we can see the oncoprint describing all the 14 patients that we found, most of them with amplifications, few of them with both amplifications and mutations. And we can see all the concurrent alterations, and we compare this with the rest of the patients that were ERBB2 negative, and we can see some differences between these two groups. MSI high, microsatellite instability high, was more frequent in patients with ERBB2 alterations. This almost reached the statistically significant. ERBB3 was more frequent. PIK3CA also was more frequent. And P53 too, and this reached a statistically significant threshold. And interestingly, KRAS was less frequent. This was only a trend, but it's interesting because when KRAS mutations are involved in solid tumors, they tend to predict a low response to target therapy. And we have many examples in colorectal cancer that KRAS alterations predict no response to EGFR inhibition and HER2 inhibition. So we can see that in this case, using the MSK classification or on KKB, ERBB2 would be a level 2B type of alterations. So because there is a standard of care treatment for this alteration, but in another indication, it will be breast cancer and gastric cancer. And ERBB2 mutations such as S310F would be level 3A. So because there is compelling clinical evidence, this case reports I was talking to you about this drug, but there is no biomarker or this drug are a standard of care for this type of alterations in any tumor. So the take-home messages is that genome-informed therapies represent real progress in cancer research agenda and treatment. However, there's no intrinsic value in the targeted approach. You need to validate them as rigorous as you would do it with any other type of therapy. And the study of low-frequency molecular alterations warrant novel and pragmatic studies, such as basket trials. However, rigorous study designs are paramount if we are going to validate this therapy for the whole population. Well, thank you very much for the time, and also thank you all my colleagues that were involved in these two studies I present, and my mentors too. Thank you.
Thank you very much, Sebastian, for your interesting talk, and we will take the questions at the end of all the talks, at the end of the session. So now we will move on to the next speaker. For me, a pleasure to present Dr. Hernan Gonzalez. He will present the talk, Genetic Classifier for the Prediction of Thyroid Nodules, a Biomedical Development Model. And Dr. Hernan Gonzalez obtained his MD, medical doctor at the Pontificia Universidad Católica, and also PhD in medical sciences in the same institution. He have a residency in general surgery, and he developed a postdoc in molecular oncology at the Anderson Cancer Center, as well as a fellowship, a clinical fellowship in head and neck surgery oncology at the Anderson Cancer Center as well. And currently, Dr. Hernan Gonzalez is an associate professor of the Head and Neck Endocrine Surgery Department of Surgical Oncology, and is director of the Biomedical Research Consortium, BMRC, at the Pontificia Universidad Católica de Chile. So please, Hernan, welcome to this Spring Cancer Symposium, and we will be happy to hear your talk.
Thank you so much, Marcela, and to all the organizing committee for having me. I think my talk can complement very well the previous talk, which was very interesting. This is an experience that has been, I will be showing, that has been performed completely in Chile. My declaration of interest, I'm a shareholder at GeneProTX, and following the previous talk, I would like to start commenting on how we envision precision medicine and oncology, where molecular diagnostics are helping us more and more select best treatments for patients, which translates into improving clinical utility and health economics of our treatment for patients. Most of these strategies are based on multi-analyte signatures, and I say analyte because it's not only genomics or mutations that drive these kind of tests of companion diagnostics, but there could be multiple types of analytes that go from proteins, metabolomics, genomics. So it's something that is growing at different levels. And obviously machine learning has become a hallmark in helping us process the data that we need in order to produce multi-analyte algorithm assays, which are the tests that today are providing more and more accuracy to predict certain treatment responses, or allowing us to select patients for treatment, or even predicting outcomes in the future. So the first thing when we develop a test of this nature is decide or choose a relevant clinical question. In my space of expertise, which is surgery, and specifically head and neck surgery, thyroid cancer is one of the most frequent tumors that we encounter. And in the initial process of evaluating a patient with a thyroid nodule, patients undergo frequently a fine needle aspiration and cytological analysis. And in 80% of the cases, you will have a response, whether it's cancer 10% or benign 70%. But we have this problem in the middle where about 20% or 1 out of 5 cases are called indeterminate. The problem with this group is that these patients have a 25% risk of malignancy, and this risk drives diagnostic surgery in many cases, which results ultimately in that up to 75% of these patients actually do not require surgery. So we need a diagnostic tool that will allow us to identify patients with benign disease and avoid unnecessary surgery. This clinical question that I'm showing here is the same kind of question you could use for any kind of drug or other kind of treatment. In this case, it's more traditional because it's a surgical procedure, but it also applies to all the drug discoveries that are ongoing today. So molecular diagnostics has become a new option to address this particular unmet medical need, where designing the test, we wish to have a test that will have a high negative predictive value that will reduce the risk of malignancy from 25% to less than 5%, which will allow us to avoid surgery and recommend follow-up for the patient. Whereas alternatively, we would expect the test to have a high positive predictive value, therefore increasing the risk of malignancy from 25 to up to 70%, which would justify recommending surgery for these kind of patients. So initially what we performed was a review and meta-analysis. And one of the questions we asked was, what is the minimum sensitivity and specificity of a test of this nature to impact patient outcome? So we determined here that with a malignancy prevalence of 25%, as I mentioned earlier, and with a negative predictive value greater than 94%, that therefore post-test risk of malignancy of less than 5%, the minimum sensitivity of the test would have to be at least 90%. In addition, to determine to have a positive predictive value of 60% or more, again, in a range of malignancy prevalence between 20 and 30%, we determined that the specificity of the test that we would find to be optimal for patient impact is 80%. So having established these statistical parameters, we sought to develop a new genomic test that would allow us to help us identify these patients that have indeterminate cytology and avoid surgical removal of the thyroid for diagnostic purposes. So we started off with a literature review and identified 18 candidate biomarkers, and we chose our platform to be real-time PCR for screening. And so what we did, we collected a prospective cohort of 114 patients, 38 thyroid cancers and 74 benign disease. And in this graph where you see the relative expression or differential expression, and the 0 represents the benign disease and the bars represent the differential expression, where you can see that some of these genes have up to 30 to 40-fold overexpression or are downregulated. So there are some of these biomarkers that seem to be interesting by themselves, but we ultimately realized that none of these biomarkers by themselves provided a sensitivity and a specificity as we needed for our test. So with the biomarkers and with the data we had, we chose machine learning strategy called multi-layer perceptive neural network to help us develop a classifier that would increase the accuracy of our test. And so we did a second study where we collected fine needle aspirations of patients, which is the actual kind of sample you need for these kind of cases, and randomly separated them into a training set and to a validation set. Trained the algorithm with using the biomarker, the 18 biomarkers we had, and the ultimate classifier we ended up was very interesting, given that it provided in the training set a 93% sensitivity and a 91 specificity, and in the validation set, which is an independent cohort, it was reproduced. So this showed that our classifier was very robust. Interestingly enough, the classifier chose only 10 out of the 18 genes, 5 of them within the area of inflammatory genes, 3 of them that are epithelial genes, and 2 genes that actually don't have any relationship with cancer, but are more stabilizing genes within the classifier. So we set up an assay in order to be able to use this in a clinical setting. And so each target gene was multiplexed with a housekeeping gene, and we developed a first version of our assay and named the thyroid genetic classifier based on a multiplex qPCR setting, which showed to have a sensitivity of 96 and a specificity of 87. So this is to this point, we had a biomarker discovery. We had achieved biomarker discovery, developed the assay format, and performed analytical validation of the assay. And now we needed to perform a clinical validation in order to show that this test really could hold this performance in a larger population. So in this year, early this year, we published 2 multi-center independent trials that were prospective, double-blinded, in 2 significantly independent clinical settings. One of the trials was performed in Chile, and the second trial, independent trial, was performed in the US in order to be able to expose the test to different clinical settings and genetic backgrounds and ethnicities. And so we collected 4,000 fine needle aspirations, 897 were indeterminate, approximately 20%. So it's in the range that you would expect. And ultimately we ended up with 270 indeterminate cases with the gold standard because we needed patients to undergo surgery in order to compare the classifier results with the valid gold standard biopsy. So just to show here, the first important thing is that we had both academic and community centers in both trials, which is important to have a representative population, and because academic centers can have worse kind of tumors, and that could generate bias in the trial. So we had a pretty reasonable spread out in terms of the patient profile. Secondly, we had broad ethnicity, which was one of the focuses of this trial, where in the US population, we had African American, Asian, and other kind of ethnicities, whereas in the Chilean trial, as you would assume, was mostly white and Hispanic. So with this profile of patients, first of all, we compared the gene expression profile using the same strategy with the PCR, where in 0 you see the benign cases, and in the bars you see the relative differential expression. And interestingly enough, you can see that both for the Chilean and the US trial, the gene expression profile is pretty much the same, where the genes that are upregulated follow the same profile, and the genes that are downregulated for both countries follow the same profile, indicating that this differences in the population does not impact this particular signature. So we first analyzed the sensitivity and the specificity of the trials individually, and you can see that sensitivity for both the Chilean and US trials was 90 and 91% respectively, and specificity was 89 and 87. Therefore, there are no statistical differences between these two studies, which allows us to pool the trial in the 270 patients, showing that we had achieved the required clinical performance that we needed in order to have a useful test for these kind of patients. So from the studies performed so far, we were able to claim that the thyroid genetic classifier demonstrates equivalent performance in 2 independent, ethnically diverse cohorts, accurately predicting benign thyroid nodules that can undergo surveillance as an alternative to diagnostic surgery. So, so far in the assay development, we had reached clinical validation, but when you want to use a test in this kind of setting of precision medicine, you need to prove clinical utility, which has to do with how physicians respond or use these kind of tests, and what is the actual net benefit for patients. And there has to be a health economics case as well in order to get coverage. So we performed a prospective single center study here in Universidad Católica, including the departments of endocrinology and surgical oncology, a prospective 12-month medium study, non-interventional. And the proposed protocol was only to include indeterminate thyroid nodules less than 3.5 centimeters and a low intermediate risk ultrasound. And I have to also mention that physicians were free to choose whether to offer the test or not to patients. So it was based on clinical appreciation, whether they thought the test would be of value to them or not. So in this period of time, 1,272 FNA samples were collected. Of these patients, 244 were indeterminate, again, in the range of 20%. Physicians decided not to test in 89 patients because they thought that the clinical assessment based on patient profile and the ultrasound provided them enough information to make their clinical decision. These patients, 33 cases did not undergo surgery, just underwent clinical follow-up, and 56 patients were recommended surgery based on the ultrasound. And 39% of these cases ultimately were malignant. Therefore, 61 cases underwent unnecessary surgery. In those patients that testing was recommended, about 2/3 of the cohort, 114 patients were classified molecularly as benign, And of these, 112 ultimately did not undergo surgery. Therefore, physicians, in fact, believed what the test result and followed recommended follow-up instead of surgery. And 2 patients underwent surgery given because they had nodule growth, and both of these 2 nodules ultimately were called benign. In the group that were molecularly called malignant, 41 samples, all patients were recommended surgery, and 88% of these cases were malignant. So basically, without the assay, the malignancy rate was 39%, and with the test, the malignancy rate was 88%, showing that in a real-world clinical setting, that the test enriches the number of cases that are malignant and avoids a very significant amount of unnecessary surgery. So finally, from a health economics perspective, we conducted in this same cohort an estimated health economics evaluation. The median cost of surgery in the public system is approximately $4,000, and the test value is $1,200. And so if all patients had undergone surgery, the cost of surgery on all these patients was estimated at $620,000. However, in these patients that underwent testing, the cost of the testing was $186,000. Of these patients, 114 were called benign, and 26% were called malignant. 2 of the patients under surveillance underwent surgery. Therefore, a total of 43 cases underwent surgery, resulting in $172,000 in cost of surgery. So the total cost of testing all patients and surgery, and only those cases that were positive, is $358,000, resulting in a net cost reduction of 42%, which supports the claim that a test a genomic test of this nature not only provides net benefit for patient management, but also has a positive health economic case. Therefore, today, with this test, we have been able to go through all the phases of development, validation of a genomic test for precision medicine, and this test has been incorporated into the American Thyroid Association management guidelines. So, given that the evidence has completed its circle. My acknowledgements to our team, our basic science team, where we did the gene discovery and bioinformatics work, Estefania, Rodrigo, Soledad, Martin Fisher, and Sergio, and our clinical trials group, where they do all the quality management, trials and patient recruitments and follow-up at different levels, which were critical in order to have these 2 trials that were conducted from Chile successful. Thank you very much.
Thank you very much, Hernan, for the very interesting talk. I remember to the audience to include the questions in the chat, so at the end we can, in the end of the sessions, we can discuss about all the topics. And so now we move to our next and last speaker. Our last speaker is Dr. Verónica Burcio. She's a senior researcher at Fundación Ciencia y Vida and Andes Biotechnologies, and also is associate professor at the Faculty of Life Science and the Universidad Andrés Bello in Santiago. Dr. Burcio, she is a biochemist from the Universidad Austral de Chile and a PhD get her PhD in molecular and cell biology and neuroscience from the Faculty of Science of Universidad de Chile. And for several years, Verónica has been working in the area of molecular basis of cancer and has authored several international patents and patent applications in cancer therapy. Her research focus on the family of non-coding mitochondrial RNAs, their involvement in cancer development, their application as molecular targets for cancer therapy, and the molecular mechanisms underlying the selective death of tumor cells by knockdown of the antisense member of this family. So with pleasure, we hear the presentation entitled Targeting Antisense Mitochondrial Non-Coding RNAs for Cancer Therapy, Lessons from Preclinical Trials. Thank you, Veronica, for your participation in this interesting symposium. And you can share your screen and maybe your audio. Veronica, can you activate the audio?
Yeah.
The speaker. Thank you so much.
Okay, sorry about that. I'm sharing my screen, I think. Can you see my screen?
Yes, very well.
Okay, I can't see it now. Sorry. Sorry. Where can I see that? No, I don't know what happened here.
It's okay. Maybe the presentation mode, if you can put in the presentation mode in the lower right of the screen. I think it's in the lower right of this of your screen.
I can't see my own screen. I'm sorry. I don't use this. Oh, okay. So I can see you now.
Yeah, now you can share your screen again.
Okay.
Yes.
Okay, share.
Mm-hmm. Exactly. Fantastic. Okay. Great. Okay. Thank you, Veronica.
Okay, so yeah, so as I was saying, I want to thank the organization for the invitation, of course. Thank you, Marcela, for that introduction also, and very happy to be in this Congress. So, well, as already stated, the title of my talk is Targeting Antisense Mitochondrial Non-Coding RNAs for Cancer Therapy, Lessons from Preclinical Models. What is this, these antisense non-coding mitochondrial RNAs? The family of non-coding mitochondrial RNAs was described by us a few years back, several years back, in which we found these transcripts that are derived from the bidirectional transcription of the mitochondrial genome. And of course, among this transcription, we will obtain the transcript of the 16S RNA, ribosomal RNA gene, both the sense version, that is, the one that actually forms structural part of the mitochondrial ribosome, and also an antisense version, which is from the transcription of the complementary strand. So we found these types of transcripts that are actually chimeric because they come from both different transcription of the mitochondrial genome. And one of those is formed by the sense 16S ribosomal RNA, which is here in blue, and it has at its 5' end a segment of the antisense transcript from the same gene. So you get this inverted repeat, which predictably forms a long double-stranded RNA structure with a single-stranded loop. So a type of a hairpin structure, if you like. And so we also found a converse version of this transcript formed by the antisense 16S transcript, and here in red, and a segment of the sense transcript, also forming the same type of secondary structure. So we termed these transcripts as sense and antisense non-coding mitochondrial RNAs. We found that in different cell statuses, proliferative statuses, when we detect these different RNAs, the antisense and the sense version, by in situ hybridization, chromogenic or fluorescent, the same, we find differential expression of the antisense and the sense non-coding mitochondrial RNAs. So in resting cells, we can barely detect just a very background level of both of them, antisense and sense, antisense here in red and sense in blue. While in normal proliferating cells, like epithelial cells or whatever, we can find a high level, a very detectable level of both the antisense and the sense transcripts. In tumor cells, however, we find we are readily able to detect the sense transcript, but we find a very background detection of the antisense version of this family. So based on this and also on other works, we deduce that the downregulation of the antisense transcripts, because there's actually more than one, is a step during tumorigenesis, carcinogenesis. How? We still don't know. And lately, recently we found a fourth phenotype in senescent cells in which it's actually the converse situation as tumor cells, where we have a high detection of the antisense and a low detection of the sense non-coding mitochondrial RNAs. But if we focus on normal cells, normal proliferating cells, and tumor cells, the only difference would actually be that normal cells have a high detectability, so a high expression of the antisense transcripts, while tumor cells have a low expression of these transcripts. So we thought of these transcripts, the antisense, as a sort of vulnerability or weakness or Achilles heel of tumor cells. And besides, considering that in all the tumor cells we have analyzed from different tissue origins, in cell lines and in tissues, we see the same pattern of expression. So we thought about knocking down the sense transcript in order to render the tumor cells somewhat like resting cells, but that didn't work out. So we tested knocking down the antisense transcript, which in tumor cells are very lowly expressed. So we actually developed an antisense oligonucleotide called 1537S, which is now called ACCDiS 1537. So when we transfect tumor cells in culture, cell lines in culture, with this antisense oligo, we knock down the antisense transcripts, and these cells eventually die. They die massively by apoptosis. And before death, they suffer a very strong cell cycle arrest. I forgot to put this closer to this because this does not happen in normal cells. So we have a very drastic lowering of proliferation, a very massive apoptotic cell death. And in the surviving cells, we see also, which are very few, we see also a strong decrease in tumorigenicity, migration, and invasion, which are, parameters for metastasis. So in developing this, this approach for visualizing a future treatment, we have tested many tumor cell lines, and in both in human and mouse. And here I am showing you just a few of the ones we have tested, such as breast cancer cells, melanoma, hepatoma, cervix cancer, prostate cancer, lymphoma, leukemia, bladder and kidney cancer. As I said, just a few examples. And we have also always observed at least a 50% degree of cell death by apoptosis, as you can see here in the blue bars. So in the green bars are just a few examples of normal cells which are not affected by the treatment. The same oligo transfected into these cells does not affect, for example, resting or activated lymphocytes or endothelial umbilical cord cells. This is also the case for mouse cells, which, as I will show you, it's the same pattern of expression as in human cells. So here are the examples of B16, F10 melanoma. There's a myeloma and a bladder cancer cell line. And in these two normal cell types, myoblasts and bone marrow mesenchymal cells, there is just a basal amount of cell death. So it doesn't affect them. And here just to show you that by several parameters, among which are the ones that I'm showing you here, we have determined that the cell death occurs through apoptosis, which is good considering the application of this treatment as therapy for cancer. So we have at least nuclear fragmentation, chromatin fragmentation, annexin 5 binding and caspase activation in all the cell lines also that we have tested with this treatment. Now at the molecular level, we have found that the treatment of knockdown of these transcripts, the antisense transcripts, generates down regulation of several key proteins. Some of these are survival factors, as I'm showing you here, such as survivin, which is a very important survival factor in cancer. It's actually the focus of many studies nowadays. So we found a strong downregulation in survivin and CIAP, which is a companion of survivin. And we also tested BCL2, which was not downregulated, and actually sometimes we see it upregulated a little bit. But we do find a change in the subcellular localization of BCL2, where in control cells, untreated, non-treated cells in control also, we find them associated to the mitochondrial cytosol. Whereas with the 1537 treatment, we find the BCL2 protein associated more to the nucleus, inside the nucleus. So, and the important thing is that inside the nucleus, BCL2 actually fulfills a pro-apoptotic function instead of an anti-apoptotic function. So we found that there is a strong downregulation, practically disappearance of the protein FKBP38 or FKBP8, as it is also called, which is a chaperone that actually brings the BCL2 protein to the mitochondria to act as an anti-apoptotic protein. So these molecular changes could account for the massive death that we're observing, but we're also interested in this other effect that we see before death, which is a very strong inhibition of proliferation. Here you can see a curve, a growth curve of MDA-MB231 breast cancer cells, and where these cells practically stop proliferating from the beginning, whereas the controls, non-treated cells, and control also treated cells don't keep growing normally. And so by Western blot, we determined that this absolute arrest in growth occurs through strong downregulation in cyclin B1, cyclin D1, CDK1 and CDK4 at least. These are the proteins that we have analyzed to date. While we see that other cyclins like cyclin A1 and cyclin E1 are unaffected by the same treatment. So this is specific for these two cyclins and these two CDKs, at least until now. And also I put survivin here again, because survivin is not only an inhibitor of apoptosis, but is also a regulator, is a cell cycle progression factor in the M phase. So it's also a protein that has to do with cell cycle. So this kind of result we have actually obtained the same result for several other tumor cell lines from other different origins, such as melanoma, actually human and mouse melanoma, other breast cancer cells, kidney, I think, and others. So it seems to be universal, one could say, phenomena. So as with cellular parameters, we have observed that after treatment, there is a strong reduction in sphere formation under non-adherent conditions, which is indicator of tumorigenic potential. So we see a strong reduction in that parameter. And also in a transwell matrix or matrix invasion assay, we also see a very strong reduction in invaded cells into the matrix. And of course, if there's a reduction in invasion, there's probably a reduction in migration. So this is a parameter that's indicative of metastatic potential. So with all these evidences, we wanted to translate this to in vivo studies, of course, with mice. So we already had the evidence that mice also express these transcripts, both as sense transcripts, which we term M, sense non-coding mitochondrial RNA, and we have found 3 antisense transcripts, which are also differentially expressed, being detectable both sense and antisense in normal cells, and in tumor cells, we only detect the sense transcript, and the antisense are practically undetectable. So the first thing we started out doing was an ex vivo treatment in which we treated cells, this murine melanoma cell line, B15F10. We treated them in vitro or ex vivo with the ASO, an oligonucleotide specific to the mouse antisense transcripts. And after treatment, we do different cellular assays, invasion assay, colony formation assays, and we count the cells, and we inject the same number of live cells by trypan blue exclusion into syngeneic mice, which in this case is C57 mice. So we also saw by the treatment ex vivo that the growth curve showed an arrest of cells treated with the specific ASO. And cell death was increased. In this cell line, we obtained between 60 and 80% typically of cell death. So also, the invasion assay showed also a strong reduction in invasive potential with the specific oligo compared to the control oligo. And of course, again, colony formation assay was drastically, or colony formation capacity was drastically reduced in cells treated with the specific ASO. So here on the side is the result of the injection of the same amount of live cells subcutaneously into C57 mice, and just a simple monitoring of the growth of tumors in these mice during in time. So we saw that in a couple of weeks, all of the tumors, all of the mice that were injected with cells treated with the control ASO or not treated, these two curves, had developed tumors, all 5 mice from each of those groups. The other mice, none of the 5 mice developed any tumor, not even up to 140 days, which we left them for that long just to see. And in all that time, none of those mice actually developed any tumors. So now we are interested in going on to in vivo studies. So again, the first in vivo studies we did were in the same model, B16F10 melanoma cells in syngenic mice, C57 mice. So we injected the mice subcutaneously with 100,000. This is what we routinely do for this cell line, 100,000 cells in the right flank of C57 cells. And around 10 to 12 days, we can see the start of the development of tumors, small tumors, which were removed by surgery. After surgery and suturing, the mice were subjected to varying numbers of injections of the ASO, either intraperitoneal or intravenous. And then we watched the growth of the tumors. And also after necropsia, we checked for metastasis. So the first result that we got was in this graph. You can see the growth of the tumors, of course, is the same for both groups, the control group in blue and the therapeutic group in red. So after surgery, of course, the tumors disappear. And after injection of several doses of the control or the therapeutic ASO, the mice that received the control ASO rapidly developed a tumor in the primary site again, in which these mice were sacrificed when they reached a little less than 1,500 cubic millimeters. But the ASO, the treatment group, the 6 mice in the treatment group actually never developed any relapse of the primary tumor in the same site. So at the end of the experiment, after necropsia, we looked at the liver and lungs, and we saw that in the control group, the livers had developed very big metastatic tumors here, and several smaller metastatic foci, while in the therapeutic group, we practically saw no metastasis on the surface of the organs. The same thing happened with lungs. In lungs, we saw in the control group, we saw many metastatic foci, some with big tumors. And in the therapeutic group, we only saw a couple of small foci in a couple of lungs, but a very strong reduction in metastasis. Another experiment that we did with this same cell line, same type of mice, was tail vein injection and checking for lung colonization after treatment. And so, as you can see in the ASO control group, there's a big, very, very, very severe colonization of the lungs, whereas with the treatment group, we saw much fewer foci in the lungs. So despite the fact that you can still see metastasis in these lungs, one could possibly attribute it. I mean, the difference with the assay we did in vivo, which is real metastasis. This is an assay where you inject hundreds of thousands of cells into the vein, and the cells directly colonize the lungs. So that could be the explanation for that. We also, with this same model, we did another approach in which, instead of using antisense oligonucleotides, we used a lentiviral approach with lentiviruses encoding shRNAs against these transcripts. And here we saw, we obtained the growth curves of the tumors measured by caliber, in which we saw a very strong reduction in tumor growth. And at the end of the experiment, the size of the tumors were much smaller than the control groups, the non-treated mice and the control SH treated mice. So this is also reflected on the tumor weight here in the graph, where the therapeutic group has much lighter tumors. Also, with the tail vein injection assay, we also saw a strong reduction in metastatic foci in the treatment group as compared to the control groups. And micrometastasis was also strongly reduced, as you can see here in the graph. And finally, overall survival was increased with the treatment group. So basically, this type of approach we did here was just another proof of concept of the antisense transcript, these antisense transcripts as molecular targets for cancer therapy. So, of course, as we tried in several types of cancer cell lines, tumor cell lines, and we found that in each and every one of the tumor cell lines from different tissues, we had the same effect, strong cell proliferation blockage and a strong massive apoptotic cell death. So we thought maybe this treatment could be kind of universal. So in this respect, we have been accumulating different models of in vivo studies, both syngenic, as I just showed you for melanoma, and xenograft studies. So here we have another model we use, which is the Renka cell line. This is a mouse renal cancer cell line in syngenic mice, which in this case are BALB/c mice. So it's the same approach. The cells are injected subcutaneously in the mice, and then the mice receive injections, in this case just IP injections through a course of time. And in this assay, we actually saw a reduction in all the tumors generated. I'm showing you here in green just the curve with the whole group of mice that received saline and the mice that received the control ASO in blue. And in red are each one of the curves of each mice in the therapeutic group. So you can see some mice developed small tumors and then they disappeared, they shrunk. Others developed mid-sized tumors and also disappeared. And this mouse developed a large tumor and it curiously also disappeared. That was surprising, actually, because coming from such a large tumor, having shrunk up to indetectability was very surprising to us. So the overall, the survival rate of these mice was actually 100%, while the control mice just survived a little under 30 days. I mean, after they had to be sacrificed. So with this same cell line in the same mice, of course, we also did orthotopic studies after the results of the subcutaneous model. So here the cells were implanted under the kidney capsule, and at 4 days injections of the ASO began, and necropsy was performed at 19 days. So here you can see, in this case, we used another ASO called 1232 sense, which is also against the mouse antisense transcripts. And you can see while the control mice on this side developed very large tumors as compared with the other kidney, the non-injected kidney. But the therapy group, in the therapy group, only one of the mice developed a not so big tumor on his kidney. So the tumor weight and subtracting the injected kidney, subtracting the non-injected kidney from the injected kidney was very different in weight. Also, micrometastasis was very much reduced, as you can see by these figures in this graph. This is another model. This is another Renka model, which is the tail vein assay, in which rapidly you can see a very strong decrease in micrometastasis, which is represented here by the graph here, and also a much higher survival rate for the therapeutic ASOs compared to saline.
Sorry, Veronica, for interrupting. We have like 3 more minutes to start the round session discussion. Sorry.
Okay, I'll hurry up. So I'm just going to show you a xenograft model. This is for xenograft breast cancer model with this cell line, which here we monitored primary tumor growth, much delayed with Andes 1537 against the human antisenses. And this is a post-surgical tumor relapse study in which we saw the primary tumor was smaller because they were receiving injections, and the relapse was much higher in the control also treated. And finally, this is the last model that we published, which is also xenograft, subcutaneous model for bladder, which we use this UMUC3 bladder cancer, human bladder cancer cell line, where we saw also a delay in the growth of the primary tumor. And here is just the apoptosis inside the tumor. And on the other side, we have a PDX model, where we saw the same tendency. So that's all in conclusion. Well, I don't think I can read them. The knockdown of these transcripts affects cancer cells negatively at different parameters. And we have done several preclinical models to show the effectiveness of proof of concept, and just show you the lab, and that would be all. Thank you very much for watching and listening. So I will stop sharing, right?
Yeah, thank you very much, Veronica, for your interesting talk. And now we invite the speakers of this session to, if they can, to activate the camera so we can go through the questions that participants include in the chat and also in the social media.
So I will be helping Marcela with the roundtable discussion because Alejandro had to leave. Maybe I could start with a question to Marcela herself.
Okay.
So this is a question by Dennis Bravo. She says some bacteria have been associated with the development of colorectal cancer. For instance, I think it's Falciparum nucleatum. Are some of these bacteria involved in the activation of the small ST2 IL-33 complex?
Oh, thank you, Denise, for the question. And that would be interesting because there are not many studies relating bacteria and this inflammatory system. For sure, if they are doing some damage in the epithelial tissue, they will definitely induce the release of IL-33. So that would be interesting to see. In some other cancer models, for example, liver fluke in cholangiocarcinoma, was seen that IL-33 is released from the epithelial cells. So that would be interesting to evaluate and correlate an association between these two elements. Thank you.
Marcela, maybe you can ask a question or two out of the
sure. Yes,
that you have in the WhatsApp there.
Yes, I have a question to Hernan from Alejandro Corbalan, that he's asking what is the ideal number of genes for the classifier analysis. In your classifier analysis, you use 10, but he said if it's more is better, or there's any criteria for this number of genes included in the analysis.
That's right. Yeah. Thank you for that question. That question is a difficult one to answer. Ultimately, it depends on what you're looking for. This classifier, when we were developing it, we were thinking about the end user, somebody that could use it relatively easy in their laboratory. More genes does not mean better clinical performance. There's a lot of these classifiers that have thousands of genes, especially through NGS, and many of these genes are just they're interrelated, and therefore they do not provide actual value from a diagnostic performance perspective. Therefore, trying to have the least amount of genes in any kind of these tests seems to be a better idea, given that from a platform perspective and a control perspective, when you're developing an assay, you have less biomarkers you need to control for, and makes it more simpler. And if you want to further develop a kit and put it into an IVD or an in vitro diagnostic kit, the less genes you have, the easier it is to manage.
Thank you. Thank you so much. There's another question from Alejandro Corbalan to Sebastián Mondaca. I would like to ask you if the access from Chile to these trials as a patient or as a clinical researcher, if you can comment.
So admittedly, it has been more difficult to conduct target therapy trials here in Chile because of the infrastructure that you need and timing. Immunotherapy trials have been easy to run here, of course, in connection with in the setting of clinical industry-sponsored trials. But we are trying to move to earlier type of trials like phase 1 and phase 2, and hopefully one day to have like a bigger portfolio in terms of target therapy trials. But I agree that definitely is one of the challenges to have more clinical trial opportunities for patients that consider genomic profiling.
There's another question to Hernan Gonzalez from Karen Dubois. She would like to ask if you did this validation separating the population by gender, and if or you can obtain similar results. She really enjoyed the talk. Thank you.
Thank you. Yeah, we did a sub-analysis on gender. It's important to know that thyroid nodules, 80% are female and 20% are men. However, in the sub-analysis of male to female, they have the exactly same performance, both on sensitivity and specificity. So there is the gender does not impact on the performance. Does that answer the question?
Yes.
Yes, thank you.
I have another question here for Marcela from Lisette. She says, since the effect of IL-33 ST2 relies on proteolytic cleavage, Could protease inhibitors be useful as therapeutic agents?
Well, very interesting question. During inflammation, there's a broad, massive release of proteases. So it haven't shown it the system is cleaved. What is known in terms of IL-33, we just published a paper in collaboration with a group in Argentina in celiac disease that IL-33 can be cleaved. Is cleaved, and it seems that cleavage of IL-33, the ligand, is more potent in activating the receptor. However, in terms of the cleavage of the receptor, that is not demonstrated yet. So can be a very interesting therapeutic target using these kind of protease inhibitors. Thank you so much. There's a question for Veronica from Karen. Why if antioxidant treatment may impact on NCMT RNase expression?
Well, short answer, we don't know. We haven't tried that yet, but it would be, I mean, it's a nice observation. It would be interesting to try that, but we don't know anything yet.
Another question here for Marcela. No, actually, sorry, it's for Hernan. Very interesting talk. And she's wondering whether the chemokines of the CCL gene family can be included as classifiers in specific types of tumors and stages.
That question, it's a really interesting question. Our sideline research has been in chemokines and in cancer. Chemokines became more important in cancer about 15 years ago when they were first described in melanoma and how they are involved in metastatic spread of melanoma and breast cancer cells. And since then, they've been actually studied in all kinds of tumors, and the hypotheses behind their participation in tumors, not only metastatic disease, but also in tumorigenesis. So these sets of proteins are not specific for thyroid cancer, but they are informative of the biology or the status of the cancer. So we have a previous publication in about a couple years ago in Oncotype that we show that that there's certain chemokines, especially CXCR3, that is related to the metastatic status of patients, and so that particular chemokine could be related to promotion of tumor spread. So they are related to tumor status, but they're not specific for tumors per se, since they are widespread gene family in the cell in the destination of cells, both in normal and tumor cells.
Thank you very much, Anna. Thank you. There's
go ahead.
There's
another question here for Veronica. How do you inject your therapeutic, I guess, ASOs or antisense RNAs? So what is the path of administration? Have you tried naked oligo administration or transfection? Or have you tried, for instance, administration via exosomes or liposomes? And another question, have you tried combination therapy?
Yeah, so for in vivo studies, we just use naked oligo. For some reason, it's not well known yet, but for some reason, naked oligos just tend to be absorbed by tissues, which is something that does not happen in vitro. So in vitro, you need a transfection agent, but in vivo, you don't. So basically, we inject the naked ASO, mainly by IP, via IP, intraperitoneal. which has, it seems to be better than IV. And we've seen, for example, absorption of a fluorescent SO in the kidney tumor, which one of the models I showed you. So in vivo, you don't need transfection. Sometimes you could do some kind of directed delivery, but we haven't done that in any of the models. And combination therapy, we have done combination therapy, but in vitro. And it seems to like help, the ASO seems to help classic types, chemotherapeutical agents. But we haven't yet focused a lot of efforts in that direction, but yeah, we have done.
There's another question to Sebastian from Andrew Quest. What is the feasibility to perform umbrella trials in Chile?
I guess the main difficulty is that here in Chile, doing genomic sequencing is not a standard of care, and that's very different compared to the US and Europe. So most of oncologists do not have exposure to this type of testing. So that's why we have not been attractive for the industry to start those type of trials here, and they prefer to do it in academic centers in the US and Europe. So that's what we are going to try to change, and to use this type of technology when is warranted in order to obtain better results for our patients and give more opportunities for interesting clinical trials.
Yeah, there's another question here for Marcela from Karen Dubois, and she's wondering whether this IL-33 signaling access might be regulated by diet.
Thank you for the question. And of course, the contents in the diet can promote the production of metabolites that can regulate the inflammatory process. And I have no background or information from the literature that can be regulate this signaling pathway. So be interesting, for example, in colorectal cancer and also inflammatory bowel disease, there's a strong dysbiosis, so can be something worth looking and investigating. Thank you.
Yeah, I think there's another talk here, another question here for Veronica. And wondering here whether antioxidants might impact on the expression of non-coding mitochondrial RNAs.
That was before I answered that one already.
Oh, sorry.
I think so.
Okay.
There are more questions from Veronica. For Veronica, from Andrew Quest. If you see amazing effects in all models, are you doing clinical trials? Any results?
We have completed one clinical trial in UCSF in San Francisco with 16 terminal patients, where we included solid tumors, non-brain solid tumors. And there was no toxicity up to the highest dose used was 800 milligrams per kilo. There was no toxicity observed. Actually 2 of those patients, terminal patients, showed stable disease over the 6 months of the treatment. One was gallbladder cancer, and the other one was pancreas. So those were actually very promising results. And now we are doing that was a phase 1A. Now we are doing a phase 1B here in Chile with which is limited to 6 types of solid cancers. but that one's ongoing and we have no information yet. So yeah, we've been doing some.
Okay, thanks. Do you have any more questions in your chat there, Marcela?
Yeah, I have the last question from Hernan to Veronica Burcio. Great talk. Any comments on toxicity in animals you've seen?
Okay, yeah, so as I was saying for the human subjects, we also, of course, in the preclinical trials, we didn't find any after necropsia, we didn't find any toxic effects on mice. Of course, mice that had only been injected with the ASO. And we published a profile of cytokines which were actually not altered. So, oh, and there's one very curious case. These were studies done by Lorena Lobos, where she treated a mouse in these studies that was injected with melanoma. The mouse recovered and all. And that mouse was actually a female mouse that was pregnant. So she had her babies, her baby mice. The baby mice grew up and they had their own baby mice, and there was absolutely no effect in that matter. So that was a really fun result. So yeah, we up to date, we haven't found like any important toxic effects, mice or human.
Okay, thank you very much. I think we're coming to an end in the session here.
Yes.
And I would, of course, together with Marcela, like to thank all the speakers today. And we look forward to seeing you all again next week, next Tuesday specifically, the 17th of November. We have 2 more sessions there, systems biology and immuno-oncology session, but led by Alejandro Corvalán, and then a nanotechnology session coordinated by Marcela Kogan. And we have a keynote speaker, Marcela Casanietz from the University of Pennsylvania. So we look forward to seeing you all again next week. And as I said, once again, thanks to all the people who presented today and all the people who asked questions. See you next week.
Bye.
Thank you very much.
Bye-bye.
Bye-bye.
Thank you.
Bye.